Life has existed on Earth for 3.8 billion years. For 3 billion of those years, almost nothing happened. Bacteria, algae, chemical silence in oceans without predators, without eyes, without fast movement. And then, in the span of 20 million years, a blink on the geological scale, life invented eyes, teeth, legs, fins, brains, predators, prey, skeletons, centralized nervous systems, hunting and fleeing behaviors. Almost all the major body plans that exist today, including yours, appeared during that period. Tonight, we dive into the most mysterious biological event in Earth's history. We look that vanished world in the face, its creatures, its inventions, its oddities, and we try to understand why it happened so quickly, so brutally, and why Darwin himself admitted it troubled him deeply. To grasp what the Cambrian Explosion is, you first have to understand the scale of the time that precedes it, and how it makes the event even more astonishing. Earth is 4.54 billion years old. Life appears in the geological record about 3.7 to 3.8 billion years ago, in the form of stromatolites, structures formed by communities of cyanobacteria, the oldest known examples of which are found in the Dresser Formation, in the Pilbara of Western Australia. These are prokaryotes, cells without a membrane-bound nucleus, without differentiated organelles, without sexual reproduction as we know it. For 2 billion years, that level of complexity is the maximum reached. 2 billion years of bacteria. The next significant event in the complexity of life, the appearance of eukaryotes, cells with a nucleus and specialized organelles, including mitochondria, occurs about 1.8 billion years ago. Some hypotheses place it earlier, around 2.1 billion years ago, but the consensus remains within that range. About 800 to 600 million years ago, the first complex multicellular organisms begin to appear in the fossil record. Not filaments of identical cells, like some algae, but organisms with different cell types having distinct functions. Life took 3 billion years to go from a bacterium to an elementary multicellular organism. The Ediacaran biota occupies the period between about 635 and 538 million years ago. It takes its name from the Ediacara Hills in South Australia, where the first well-preserved specimens were described by Reginald Sprigg in 1946, although the significance of his discovery took years to be recognized. These organisms are soft-bodied, without hard parts, preserved in fine sandstones or fine-grained sediments under rare conditions. They are found today on every continent, in about 20 major sites. What stands out about the Ediacaran biota is its fundamental strangeness. Dickinsonia, one of the most studied organisms, is an oval, segmented form that can reach 1.4 meters in length, lacking a mouth, an anus, eyes, or limbs. Its phylogenetic classification was debated for decades. A 2018 study published in Science by Ilya Bobrovsky and colleagues detected lipid biomarkers, cholesterol, in Dickinsonia fossils, confirming its belonging to the animal domain. But to which group exactly? The debate continues. Charnia, Tribrachidium, Fractylophusis. Other members of this biota display triradial or fractal symmetries that do not match any living animal body plan. These are evolutionary experiments with no recognized direct descendants. Most of them disappear at the Precambrian-Cambrian boundary, about 538 million years ago, replaced by something radically different. The geological context immediately preceding it deserves to be laid out. Between about 720 and 635 million years ago, Earth went through one or more episodes of extreme global glaciation, what is called snowball Earth. Geological data, glacial deposits at equatorial latitudes, diamictites on all continents, suggest that glaciers reached the tropics, perhaps covering the oceans with a layer of ice a few hundred meters thick at low latitudes. Life survives in refuges, probably near submarine hydrothermal vents, in waters kept liquid by geothermal heat, and perhaps in equatorial zones where the ice was thinner and where photosynthesis remained possible under low light. The melting of these massive glaciations, driven by the buildup of volcanic CO2 in the atmosphere in the absence of active carbon sinks beneath the ice, releases considerable quantities of nutrients, phosphorus, iron, various minerals, into the oceans. This sudden fertilization creates new chemical conditions, potentially favorable to biological innovation. The hypothesis that snowball Earth functioned as an evolutionary stressor, forcing the selection of more efficient metabolisms and survival strategies, and then as a catalyst for post-glacial diversification, is widely discussed in the literature. The Precambrian-Cambrian boundary, 538 million years ago, is one of the sharpest in the entire geological record. Below it, soft-bodied organisms, without hard structures, without recognizable eyes, without body architecture reminiscent of modern animals. Above it, an explosion of forms, skeletal structures, and body plans recognizable as the ancestors of present-day groups. This is not a gradual progression. It is a rupture. The first major biological innovation of the Cambrian is also its most consequential: the eye. Trilobites, one of the most abundant and best fossilized groups of the Cambrian, possess some of the first complex compound eyes known in the fossil record. Their eyes are built not from proteins like vertebrate eyes, but from calcite, birefringent calcium carbonate, cut into individual prisms with an optical precision comparable to that of a modern optical crystal. Each prism functions as an individual lens. Thousands of these prisms form a faceted eye capable of detecting movement across a wide field of view. Some trilobite species had holochroal eyes, with thousands of contiguous lenses. Others had schizochroal eyes, with lenses separated by a corneal surface, whose optical properties have been analyzed by physicists as the equivalent of systems correcting spherical aberrations. The appearance of functional eyes in an ecosystem that lacked them represents a radical change of rules. To see means detecting prey at a distance, directing movement toward it, anticipating its trajectory. To see also means detecting a predator at a distance, fleeing before being attacked. From the moment an animal in an ecosystem can see, the selective pressure on all the others changes instantly. Invisibility, protection by armor, swimming speed, the ability to burrow, all these traits immediately become advantageous. The evolutionary arms race is launched. Andrew Parker, a biologist at the University of Oxford, formalized this idea in 2003 under the name light switch theory. In his hypothesis, the appearance of the first functional eyes is the trigger of the Cambrian Explosion. By creating intense selection in favor of defensive and offensive traits, the eye would have generated an evolutionary acceleration in all directions at once. The other biological innovations of the Cambrian follow in sequence and respond to one another. Hard mouth parts, mandibles, chitinous teeth, grasping structures enable active predation on hard-parted prey. Skeletons and shells, built from limestone, calcium phosphate, silica, provide protection against these new jaws. Jointed limbs, the appendages of arthropods, the parapodia of annelids, enable diversified modes of locomotion, active swimming, walking, burrowing, climbing, centralized nervous systems with a cephalic ganglion, nerve cords, sensory-motor connections, make it possible to integrate sensory information quickly and produce complex, coordinated behaviors. In 20 million years, life simultaneously invents vision, active hunting, rapid escape, structural defense, and the rudiments of what one might call behavior. The Cambrian bestiary is populated by creatures, some of which have no recognizable descendants today. Anomalocaris, strange shrimp in Latin, is the apex predator of its ecosystem. Reaching up to a meter in length, it is by far the largest animal of its time. It swims by undulating soft lateral flaps, like a modern ray. Its mouth is a circular structure of articulated hard plates, capable of crushing armored prey. Its two front grasping appendages, lined with teeth, seize and manipulate prey. Its stalked compound eyes, known from exquisite specimens from the Burgess Shale and Chengjiang, are among the most complex in all Cambrian fauna. A paper published in Nature in 2011 by Peter Van Roy and his collaborators identified isolated fossil eyes of Anomalocaris bearing up to 16,000 facets, visual acuity comparable to that of modern dragonflies. Hallucigenia owes its name to its first interpretation, which turned out to be wrong in two ways. Simon Conway Morris, in 1977, reconstructed it upside down. What he took to be legs were in reality dorsal spines, and what he took to be dorsal tentacles were its true tubular legs, and backwards front to back as well, mistaking its tail end for its head. It took decades and better preserved specimens from Chengjiang to establish the correct morphology of this animal. Hallucigenia belongs to the lobopod group, distant relatives of modern tardigrades and onychophorans. Opabinia, 5 compound eyes and a prehensile proboscis ending in a claw, is so bizarre that at the 1972 meeting of the Paleontological Association, when Harry Whittington presented it to the audience for the first time, the assembled paleontologists burst out laughing. Not in mockery, out of astonishment. This creature resembles nothing living or fossilized that was known. Its place in the tree of life remains debated. Perhaps a basal position among arthropods, perhaps an entirely separate lineage. Picaia gracilens, by contrast, is an unassuming animal, 5 centimeters long, resembling a flatworm. But its significance may be the greatest of the entire Cambrian. It possesses a notochord, a rigid longitudinal rod that in later animals will give rise to the vertebral column. Picaia is a cephalochordate, a member of the group that today includes Amphioxus, and a serious candidate for the title of ancestor of all vertebrates. If this interpretation is correct, this little insignificant worm swimming in the shallow Cambrian seas as the grandfather of all fish, amphibians, reptiles, birds, and mammals, including all the humans who have ever existed. The window onto this world is provided primarily by 2 exceptional fossil deposits. The Burgess Shale in Canadian British Columbia, discovered by Charles Walcott in 1909 and dated to about 508 million years ago, has yielded thousands of specimens with extraordinary preservation. Anoxic conditions on the seafloor prevented bacterial decay of soft parts, preserving not only carapaces and shells but also muscles, intestines, eyes, and sometimes even stomach contents. The Chengjiang Shale in Yunnan, China, discovered in 1984 by Ho Xiangguang, is slightly older, about 520 million years, and even richer in soft-bodied species. Why did such an explosion of complexity occur at that precise time, after 3 billion years of relative stagnation? The question is one of the most debated in paleobiology. Darwin himself was troubled. In On the Origin of Species, he devotes an entire chapter to what he calls the most serious difficulty against my theory: the sudden appearance in the fossil record of major animal categories without visible intermediate forms below. His answer was that the fossil record was incomplete, that intermediate forms had existed but had not been preserved, either because they were soft-bodied or because the corresponding sediments had not been found. This explanation remains partly valid. Soft-bodied organisms fossilize far less readily than those with hard parts, but it does not fully resolve the mystery of the apparent speed of diversification. The first major hypothesis is oxygen. The Great Oxidation, 2.4 billion years ago, enriched the atmosphere with O2 produced by cyanobacterial photosynthesis. But atmospheric and oceanic oxygen continued to rise gradually throughout the Precambrian. In the Cambrian, geochemical studies, notably analyses of sulfur isotopes in marine sediments and proxies for iodine oxidation, suggest that oxygen levels in surface ocean waters reached values close to 10% of the modern atmosphere, perhaps more, precisely around the Precambrian-Cambrian boundary. Complex aerobic metabolisms, which yield much more energy per unit of glucose than anaerobic metabolisms, become possible at these concentrations. Biological complexity, with its hearts, powerful muscles, and energy-hungry nervous systems, requires oxygen. Perhaps life was simply waiting for the fuel to become available. Andrew Parker's light switch theory, already mentioned, proposes the eye as the trigger. A single evolutionary event, the emergence of a photoreceptor directional enough to form an image, triggers a cascade of selections in favor of mobility, defense, attack, faster neural processing. This theory has the merit of explaining the suddenness of the explosion and its coherence. The entire Cambrian bestiary can be explained as a response to the visual arms race. Plate tectonics offers a third avenue. In the Cambrian, the fragmentation of the supercontinent, Rodinia created immense expanses of shallow epicontinental seas, seas a few dozen to a few hundred meters deep, warm, well-lit, rich in nutrients supplied by the erosion of newly exposed continents. These coastal environments are particularly favorable to rapid evolution. A great diversity of habitats and ecological niches creates a great diversity of selective pressures. The more available niches there are, the more evolutionary innovations that exploit them multiply. The discovery of Hox's genes in the 1980s added a molecular dimension to the mystery. Hox's genes are master regulatory genes, genes that control the expression of other genes, organizing the body plan of the developing embryo. They are present in almost all bilaterian animals, with remarkable conservation across hundreds of millions of years of evolution. The same family of Hox's genes controls development of the anterior-posterior axis in the fruit fly Drosophila, the mouse, and the zebrafish. Mutations in these genes can produce spectacular transformations of the body plan. The famous four-winged fly of Edward Lewis, legs in place of antennae. Hox's genes existed before the Cambrian. Phylogenomic analyses indicate that they were present in Precambrian organisms. What they represent is a genetic toolbox that, once in place, allows rapid morphological innovations by modifying regulatory parameters rather than by inventing new genes. The duplication and divergence of Hox's genes during the Cambrian would have enabled an explosion of body plan variations in a relatively short time on the evolutionary timescale. The current synthesis is that the Cambrian explosion is probably multi-causal. Oxygen provided the necessary metabolic energy. Hox's genes provided the molecular architecture, enabling rapid morphological innovations. Geology provided favorable environments, and the eye, or another sensory innovation triggering an arms race provided the selective pressure that sustained the runaway process. These factors are not alternatives. They likely combined and reinforced one another to produce an event that no single factor can account for. The Cambrian explosion did not triumph indefinitely. About 443 million years ago, the first major mass extinction in animal history, the Ordovician-Silurian extinction, wiped out about 85% of marine species. Massive glaciations of Gondwana, sea level fall, disruption of ocean cycles destroyed most of the Cambrian and Ordovician marine fauna. Anomalocaris, Hallucigenia, Opabinia, Wewachsia erased forever, with no identifiable descendants. But the fundamental body plans invented in the Cambrian survive through the lineages that bear them. Vertebrates, descended from cephalochordates like Pikaia, and from early Cambrian vertebrates like Millicunmingia discovered at Chengjiang, will pass through 5 successive mass extinctions, including the most famous 66 million years ago, which eliminates the non-avian dinosaurs. Arthropods, whose trilobites will die out in the Permian extinction 252 million years ago, will produce the insects, crustaceans, and spiders, which today represent the majority of known animal species. Mollusks, echinoderms, annelids, all of Cambrian origin, populate the oceans today. The chain that links a 5-centimeter worm in a Cambrian ocean to a human being watching a video on the internet has been unbroken for 520 million years. Through 5 mass extinctions, dozens of glaciations, continental drifts, asteroid impacts. Pikaia survived all that, and its descendants include all the vertebrates that have ever existed. What the Cambrian tells us about evolution goes beyond the simple history of animal diversification. It illustrates what paleontologists Stephen Jay Gould and Niles Eldredge formalized in the 1970s under the name punctuated equilibrium theory. Evolution does not occur at a constant, gradual pace, but in long periods of relative morphological stability, punctuated by episodes of rapid change. The Cambrian is the most spectacular example of this principle in the geological record. The question of Cambrian body plans not yet discovered remains open. The Burgess Shale and Chengjiang have been excavated for more than a century, yet new species continue to be described every year. New techniques, X-ray microtomography for 3-dimensional imaging of fossils without physical preparation, chemical analyses of biomarkers preserved in sediments, phylogenomics of living species to reconstruct the tree of life, open unprecedented perspectives. It is possible that entire lineages disappeared without leaving descendants or recognizable fossils in known deposits. For the search for extraterrestrial life, the Cambrian explosion raises a fascinating question. If life typically takes several billion years to go from simple cells to multicellular complexity, and then explodes in diversity once certain conditions are met, a planet with microbial life for 2 billion years could be on the verge of its own Cambrian. The ocean moons of the outer solar system, Europa around Jupiter, Enceladus around Saturn, could harbor life in their internal oceans. If so, that life may still be at a pre-Cambrian stage. Microbes, not animals. We look for civilizations. Maybe we should look for trilobites. 3 billion years of patience. 20 million years of creative frenzy. Eyes, teeth, brains, skeletons. Everything invented in the blink of a geological eye. 5 mass extinctions weathered. And at the end of that chain, you watching this video. The Cambrian explosion does not tell us that evolution is unpredictable. It tells us that it waits, that it accumulates the conditions, oxygen, regulatory genes, favorable geology, just the right selection pressure, and that when everything is ready, it can change everything in a geologically instantaneous time. The next time you see an eye, yours, a cat's, an insect's, a cephalopod's, remember, it all began there, in an ocean that no longer exists. 520 million years ago, something saw for the first time, and the world was never the same again.
At the beginning of this vast geological adventure, we dive into a period that spans from about 538.8 million years to 521 million years ago. Specialists call it the Terranuvian and consider it the first era of the Cambrian, starting just after the end of the Ediacaran. It's a time marked by mostly soft-bodied organisms, often lacking real skeletons. In this time, the Terranuvian sets the stage for a massive diversification of species. Although spectacular forms of life appear later, this era lays the groundwork for the rise of biodiversity, which would reach its peak later in the Cambrian. Let's focus on these fascinating beginnings, where the first mineralized forms and the first burrowers already signal a turning point in marine life dynamics. In this context, the biological boom is often mentioned. Although it's not yet a dazzling explosion, it's a key moment when organisms begin to make hard parts. To better visualize it, imagine a world covered with shallow seas, under a climate warmer than ours. At that time, the emerging lands are still barren, and coastal areas, unstable due to tectonic movements, vary from continent to continent. Oxygen, well present throughout Earth's history, begins to gradually increase in ocean waters thanks to photosynthesis, carried by plankton and microscopic algae. This change in marine chemistry sets the stage for upcoming biological innovations, which will mark the Cambrian rise. To better understand the history of the Terranuvian, we subdivide it into two distinct timings. First, the subperiod called the Fortunian, located between about 538 million years and 529 million years ago. Then, what is called Stage 2, or the second stage, which spans from 529 million to 521 million years. Although these terms are technical, they help describe the chronologically encountered fossils and environments. We will therefore mentally travel through these two subperiods to explore the evolution of living beings as new behaviors and body structures emerge. Let's start with the Fortunian. For a long time, paleontologists sought to precisely define the base of the Cambrian. They finally established it at a point of reference, the first occurrence of Treptichnus pedum, a unique trace fossil chosen as a marker. Treptichnus pedum is not a classic fossilized animal, but the imprint left by a burrowing organism that systematically dug through sediments. This trace, in the form of galleries or tunnels, reveals a new behavior. Instead of just stirring the surface of the seafloor, the probably worm-like animal burrowed a deeper and more methodical path. This trace is significant because it shows an advance in motor and sensory complexity, marking an evolutionary shift. The animal world at the beginning of the Cambrian begins to invest in sediments, multiplying ecological interactions, hunting, fleeing, searching for food within marine substrates. In the Fortunian, a major innovation appears with traces of hard structures on previously soft organisms marked by the small shelly fossils. These hard elements, often of millimeter size, include tubes like those of Tubulospina or microbilaterian shells like those of the Micrina genus, which could be the first bilaterian organisms to possess mineral protection. These micro-remains, often found mixed with other fossils, demonstrate the emergence of shells and rigid stems to support, defend, or anchor these organisms. This phenomenon of embryonic biomineralization reveals an adaptation process in the Cambrian seas, where competition for survival becomes increasingly intense. Walking through the tropical plateaus of Australia more than 530 million years ago, we discover a geological curiosity of the Fortunian, the first calcareous archaeocyath reefs. These marine organisms, now extinct, were similar to primitive sponges. Imagine calcareous cones or cylinders, sometimes porous, assembled in colonies and forming reefs in the shallow, sunlit areas of the continental shelf. At the time, these reefs, though smaller than modern barriers, already served as refuges for many creatures and retained sediments. Thanks to the microalgae and cyanobacteria that clung to them, they created biological oases. Ecosystems are gradually becoming more complex, illustrating a key principle of marine life. The creation of biological reliefs generates ecological niches, promoting species diversity. The Fortunian is distinguished by a slow rise in complexity, construction of microshells, establishment of rudimentary reefs, and the emergence of more adventurous burrowers. Environmentally, global temperatures are higher than today, averaging over 20 degrees Celsius in surface waters. A much higher atmospheric carbon dioxide level creates an enhanced greenhouse effect and a higher sea level. The arrangement of the continents favors the formation of shallow tropical zones, ideal for archaeocyath reefs. The ocean is gradually becoming more oxygenated, albeit modestly. This planetary change, even if small, supports the rapid development of innovative faunas. In stage 2 of the Terranuvian, around 529 million years ago, a major advancement occurs with the appearance of the first trilobites, considered icons of the Paleozoic. These arthropods with segmented shells and complex eyes move along the seabed, and their fossils are present on all continents. Their body, divided into 3 lobes and articulated rings, is characteristic. Found in regions like Morocco, Siberia, or China, the trilobites show the rapid dispersion of marine life. Classified among the red lichida, a primitive group, they will quickly diversify, illustrating the vitality of the Cambrian seas. The emergence of the first trilobites raises 2 major questions. Why do arthropods thrive so rapidly, and what environmental changes enabled this evolution? Their articulated shell offered protection against predators and facilitated locomotion in varied environments, while serving as a muscular support for their movements and search for food. On the environmental side, geochemical analyses show a gradual increase in oxygen levels, although the seas are not yet fully oxygenated in depth, but sufficiently for a more active metabolism. Although isotopic excursions are not as marked as later in the Cambrian, some rock formations show a slow evolution of oxygen content, indicated by isotopic signals. Thus, stage 2 of the Terranuvian offers a world of discovery with a fauna that is perfecting itself and an environment in mutation. Paleontologists and geologists notably travel to Morocco to study these early trilobites and the sediments in which they were fossilized. In Siberia, large fossil accumulations, exposed by geological sections, show almost continuous stratigraphic sequences that tell the progressive establishment of the fauna. In China, the reading of rock layers confirms the simultaneous presence of different red lichida groups, suggesting an abundance of ecological opportunities. The Cambrian is a time of upheaval and changes that are inscribed in the rock strata. To reinforce the symbolic importance of the Terranuvian, the Fortune Head Cliff, located on the coast of Newfoundland, Canada, is a key site for discussing the base of the Cambrian. It serves as the starting point for debates about the beginning of the Cambrian because it clearly shows the transition between the Ediacaran and the Fortunian. The last traces of the Ediacaran fauna, followed by the appearance of Treptichnus pedum, mark the beginning of the Cambrian. Scientists from around the world take samples, analyze the chemical composition of the layers, and confirm the dating, making this site a perfect example of the debate over the definition of a geological boundary. To fully grasp the significance of this anecdote, let us imagine ourselves at the base of the cliff. We would observe an accumulation of sandstone and shale that might seem ordinary to the untrained eye. Yet, upon closer inspection, one can distinguish the sinuous imprint of a burrowing tunnel, Treptichnus pedum. Paleontologists marvel at this fossil track, marking the beginning of a new era where life, now more mobile, begins to perform genuine engineering work within the sediments. This is why Fortune Head has become the historical site of the Cambrian's Kilometer Zero, a major milestone in Earth's history. Isotopic excursions during Stage 2 enlighten us on an important point. Carbon isotopes are measured in carbonates or organic-rich shales, and fluctuations between carbon-12 and carbon-13 reveal changes in oceanic chemistry. A positive isotopic excursion indicates strong carbon storage or a chemical fluctuation, while a negative excursion signals disturbances, such as a massive release of carbon. At the beginning of the Cambrian, these variations are less marked than in other periods of the Paleozoic, but an enrichment in oxygen seems to be occurring, preparing the environment for future upheavals. These analyses are essential for understanding the role of oxygen in the proliferation of animal life. What we know about the Fortunian and Stage 2 is striking. Life equips itself with hard parts. The first reef communities appear, and some animals probe the superficial layers of sediment for food or protection. The first trilobites mark the beginning of the rise of arthropods in the seas. Although the greatest Cambrian biodiversity explosion will occur later, the Terranuvian is the foundation upon which these evolutions rest. From a paleontological point of view, the Terranuvian is sometimes considered discrete compared to the rest of the Cambrian because its fossils seem smaller and less spectacular than those found in sites like Chengjiang or the Burgess Shale. Yet, the Terranuvian is a genuine laboratory of evolution, a period of experimentation marked by several fundamental innovations. Think of directional burrowing, searching for food in sediment, but also digging galleries for protection or storing reserves. Think of programmed biomineralization, a survival advantage in increasingly competitive seas. Think of the appearance of the first calcified reefs, genuine mini ecosystems ahead of their time. These steps make the Terranuvian essential for understanding the dynamics of life on Earth. If we project ourselves into these ancient oceans, we imagine a clear and shallow underwater landscape, bathed in sunlight, with a sandy bottom, probably rich in organic particles. Filamentous algae mats ripple near the surface, while fields of cyanobacteria, whose presence can be deduced by the dome-shaped stromatolites typical of Cambrian platforms, spread. In this setting, a few emergent tubulospina tubes are seen, remnants of small tube-dwelling animals capable of partially detaching from the substrate. Scattered macrina shells are found, perhaps from organisms fragmented by low hydrodynamics. Further offshore, colonies of archaeochiathids stand and filter the water, creating calcium mounds where other microorganisms gather. All this sedimentation, seemingly silent, gradually records every detail of the evolution. Tectonic forces shift the plates, creating proto-continents, and these movements influence the distribution of tropical areas, shallow seas, and ocean currents. Nutrient inputs, resulting from the erosion of the Earth's crust, vary by region, creating conditions favorable to the accelerated development of certain faunas, while elsewhere, life remains rarer. This is how geographical disparities in the fossil record are formed. Some groups abound in Morocco or China, but not necessarily in other regions where conditions did not favor their preservation. Every fossil discovered in the rocks of the Terra Nuvian enriches this picture. Although modern methods like 3D tomography or spectroscopy are more often applied to richer deposits, certain techniques allow for the analysis of Fortunian fossils from new perspectives. We can reconstruct the original mineralogy, determine if the shells are mainly phosphate or calcium carbonate, and understand their formation. These studies reveal that life experimented with various approaches to create hard parts, a mix of proteins and minerals, a thin protective coating, or external particles as a shield. This shows the almost unlimited creativity of evolution. Thus, the Terra Nuvian cannot be considered a simple discrete transition. It marks the moment when marine life becomes more dynamic, with behaviors like directional burrowing and the production of skeletons and shells. The appearance of the first trilobites in the stage 2 layers heralds not only the rise of arthropods, but also the emergence of more complex trophic chains with increasingly specialized predators and prey. If one were to retain a strong symbol of this era, it would be the trace Treptichnus pedum. It symbolizes a turning point in how organisms interact with their environment. Conceptually, this represents a change of scale in the shaping of sediments. Other indicators confirm that life is becoming more dynamic, evolving towards forms capable of exploration and the invention of new habitats. The archaeocyathid reefs reflect this, creating new spaces where other creatures can settle or hide. During the second major division of the Cambrian, called Series 2, a major event occurs, the visible explosion. Extending from 521 to 509 million years, this period is marked by a rapid diversification of life forms and the appearance of more complex creatures. To better understand, this stage is divided into two segments, Stage 3 and Stage 4. Each stage offers remarkable fossils and lessons about the evolution and complexity of life in the primitive seas. Stage 3, from 521 million years ago, plunges us into a marine world in full biological turmoil. The seas are teeming with an increasing number of increasingly specialized species. We observe the emergence of animals better organized anatomically, with armors, shells, and more developed sensory organs than those of their predecessors. This sophistication allows for new behaviors, notably in feeding and predation. Organisms no longer simply slip through the mud or passively filter their food. Some become hunters, others develop adaptations to defend themselves or better locate their resources. The ocean becomes the theater of an arms race, where prey and predators mutually influence their evolution. One of the most emblematic sites of this Stage 3 is the Chengjiang Lagerstätte in China's Yunnan province. Dated to about 518 million years ago, this deposit has yielded fossils of exceptional quality. Fine sediments have preserved not only shells and carapaces, but also more delicate details such as muscles, gills, digestive tubes, and even the beginnings of sensory organs. This rare preservation offers paleontologists a unique opportunity to study the subtleties of animal morphology at the beginning of the Cambrian. In the Chengjiang deposit, the great predator is the arthropod Anomalocaris, whose name means strange shrimp. Although it was compared to a giant shrimp, Anomalocaris is not a modern crustacean, but an ancient predatory arthropod with appendages to capture its prey. With its circular mouth and elongated body, it swam in search of less robust prey. This creature testifies to the complexity of animals in the Middle Cambrian. The fossils of Chengjiang also include chordates, such as Milocunmingia, which possesses a notochord, a post-anal tail, and gill slits. This discovery shows that the features of future vertebrates were already forming in the Cambrian. Leaving China, we set course for Greenland, where another remarkable site is located, Sirius Passet. Dated between 518 and 515 million years ago, it falls within the same time range as Chengjiang. However, Sirius Passet is distinguished by its location in a former polar or subpolar zone. This suggests harsher environmental conditions and theoretically cooler temperatures than in tropical regions. The fossils are well preserved there, even though they are sometimes more difficult to extract due to the modern icy climate. Among the main specimens discovered at Sirius Passet is a stem worm of the genus Priapulites, often compared to current priapulid worms. These worms, described as ambush animals, were probably equipped with a mouth surrounded by rigid structures to pierce or grasp organic matter. Their presence at this stage indicates a more elaborate predatory activity, reflecting the global movement of intensifying interactions in the food chain. In the seas of Stage 3, predation becomes a key factor in evolution, with organisms equipping themselves with defensive or offensive devices, thereby reorganizing biodiversity around these unprecedented selective pressures. In these ancient environments, fauna diversifies around key axes: segmented-bodied arthropods, armed or unarmed worms, early chordates, sponges, and other filtering organisms. Algae and other forms of aquatic vegetation, although less spectacular due to their reduced preservation, play a key role as primary producers. They form the base of marine ecosystems, providing food that sustains the entire food chain. The sediments of Chengjiang and Sirius Passet testify to an active world where predators, prey, and scavengers coexist in more competitive survival relationships than in the early Cambrian. We witness an increasingly complex structuring of these ecosystems, heralding an even more explosive evolution. We continue our journey into Stage 4, which extends from 514-509 million years ago. The burst of diversity initiated previously continues, but the focus shifts to perfecting traits that have already appeared. Researchers note an intensification of visual capabilities in some predators, new motor adaptations, and a marked evolution of organisms occupying various ecological niches along the seabed and in the water column. One of the reference sites for Stage 4 is the Emu Bay Shale deposit in Australia. Dated to about 514 million years ago, it is renowned for the quality of fossil preservation, allowing the distinction of fine details. Paleontologists decipher anatomical clues frozen in the sediments there. Among the discoveries is the famous predator Anomalocaris, already known at Chengjiang. The big surprise lies in the structure of its compound eyes, which contain about 24,000 lenses, a remarkable number for the time. These eyes, composed of numerous ommatidia, offer a wide field of vision and an ability to quickly detect movements. This visual sophistication is a decisive asset for hunting. Thanks to its particularly keen eyes, Anomalocaris could spot and pursue its prey with great efficiency. From an evolutionary standpoint, This means that some lineages developed, less than 10 million years after the beginnings of the Cambrian, advanced sensory abilities. This ability to perceive the environment from multiple angles gives the predator an advantage and stimulates the emergence of defensive systems in its prey. Facing such a hunter, targeted animals must camouflage themselves, equip themselves with robust shells, or improve their own vision. The complex eyes of Anomalocaris perfectly illustrate this dynamic of biological innovation from the visible explosion. In the same time frame, another fossil attracts the attention of paleontologists, Shishania aculeata, discovered in 2024 at a site dated to about 514 million years ago. The specialized press, Reuters, announced this discovery as one of the oldest forms of proto-mollusk. The term mollusk refers to a group of animals typically possessing a muscular foot, a visceral mass, and often a shell secreted by a mantle. Modern examples include gastropods, snails, bivalves, mussels, clams, and cephalopods, octopuses, squids. The existence of Shishania aculeata sheds light on the origin of mollusks, showing that in the Cambrian, organisms related to mollusks already possessed a relatively well-defined muscular foot. This foot, characteristic of mollusks, is used for moving by crawling or burrowing on marine substrates. It is believed that Shishania aculeata also had protective spines, which would improve its chances of survival in a world where predation was common. For many researchers, this specimen links simpler life forms to the more evolved mollusks of the later Cambrian, demonstrating how these lineages evolved and diversified their locomotion and defense strategies. During this stage 4, an episode of local extinction affects the archaeocyaths. These organisms, often compared to sponges or an extinct group of reef builders, had porous calcareous walls. In the early Cambrian, the archaeocyaths played a crucial role in the formation of primitive reefs, occupying shallow marine areas and building significant calcareous structures that housed other forms of life. In stage 4, nearly all archaeocyaths disappear locally, probably due to environmental upheavals such as sea level variations, changes in water chemistry, or increased competition with new lineages of sponges and microorganisms. Cyanobacteria forming microbial mats and siliceous sponges create new reefs more adapted to changing conditions. These sponges, more resistant to variations in acidity and temperature, take over where the archaeocyaths disappear, reshaping the reefs. This episode shows that Cambrian life was not stable, with selective extinctions and ecosystem adjustments well before future massive crises. Stage 4 confirms and amplifies the idea of a true visible explosion, where groups that appeared earlier refine their adaptations or develop decisive innovations. Discoveries from the Emu Bay shale site show how vision and predation played a crucial role in the evolution of Cambrian faunas. The most proficient organisms in sensory detection and locomotion access more food and can cope with more competitors or predators. This period is not only marked by the appearance of new species, but also by localized disappearances, like those of the archaeocyaths, once masters of reefs, replaced by cyanobacteria, sponges with siliceous skeletons, and other reef builders. This shows the great responsiveness of the biosphere to environmental fluctuations and competing innovations. When a group becomes too specialized or out of sync with changing conditions, it gives way to newcomers better adapted to exploit new opportunities or better protected against emerging threats. If one compares Chengjiang of Stage 3 to Emu Bay of Stage 4, there is a continuity with animals like Anomalocaris, but also a dramatic enhancement of sensory and muscular capabilities. Marine environments are populated by increasingly diverse faunas, revealing almost all the main invertebrate lineages that will dominate later ecosystems. The few million years separating these stages are therefore crucial for forming a teeming bestiary. This phase is often seen as the heart of the Cambrian radiation, where most of the organizational plans of modern animals already appear as prototypes. The fossils of these two stages show how innovations follow one another to create the diversity of Cambrian marine life. Although our focus is on animals, it is essential to consider the impact of geological and climatic upheavals on the success or decline of certain groups. In the Cambrian, the supercontinents are arranged differently, mainly in the southern hemisphere and often in the form of fragmented continents. Marine currents, sea level variations, and oxygen supply shape living conditions. In the shallow seas, burrowing, nutrition, and locomotion develop, as the loose sediment allows small animals to hide and dig. The visible explosion also reflects a favorable geodynamic context. The waters are warm, and oxygen begins to circulate more in certain areas. This supports the emergence of more energy-intensive life forms, such as robust predators with complex locomotion. In these conditions, the food chain evolves profoundly. At the base, there are photosynthetic microorganisms, such as unicellular algae and cyanobacteria, thriving in well-lit waters. Primary consumers, like some grazing trilobites, feed on these microbial mats. Then come the superpredators, like Anomalocaris, attacking herbivores or other less defended animals. This structuring into trophic levels consolidates and stabilizes during Stage 4, thanks to improved predation and defense mechanisms. The visible Cambrian explosion is thus not only an explosion of morphological diversity, but also the rise of stratified ecosystems, where each level feeds the next while undergoing pressures from its predators. The key points of Series 2 first include confirmation that Anomalocaris, present in Stage 3, becomes a more efficient predator in Stage 4 thanks to improved vision. Then, whether it's the microscopic analysis of Anomalocaris' eye or the study of the muscular foot of Shishania aculeata, we can paint an increasingly precise picture of marine life over 500 million years ago. This understanding reveals the early ingenuity of evolution, and modern organisms, including humans, owe some of their fundamental characteristics to these ancient innovations. When we talk about heritage, we often think of the transmission of information or structures from one generation to the next. In the context of Series 2 of the Cambrian, this heritage is manifested by the persistence of certain traits, like the developed vision or the modular body forms of arthropods, and the emergence of foundational groupings. Much of the current biodiversity roots back to these early evolutionary experiments. The local disappearance of the archaeocyaths reminds us of the principle of natural selection. Some lineages disappear, while others thrive and take over. We already see at this stage the fundamental forces of evolution: diversification, speciation, extinction, and ecosystem renewal. In Stage 3, the multiplication of life forms, the discovery of primitive chordates, and the flourishing of marine predators illustrate the diversity of this phase. In Stage 4, the visual power of certain arthropods asserts itself. Mollusks appear with Shishania aculeata, and the reconfiguration of reef communities shows the fragility of archaeocyaths, replaced by more opportunistic groups. In short, the visible explosion is not just a poetic expression to describe the profusion of fossils, It is also the reality of a time slot where marine life, propelled by new mechanisms of predation, sensory advancements, and defense strategies, diversifies at an unprecedented rate. The deposits of Chengjiang, Sirius Passet, or Emu Bay still allow us to reconstruct these original scenes and understand how current biodiversity originates from these ancient, rich, and stimulating waters. At the end of this period, the Cambrian has already established its foundations. Competition, specialization, and adaptation mark a world in perpetual motion. It is in this crucible that much of the complexity of animal life, of which we are distant heirs, was forged. Throughout the history of life on Earth, it is difficult to imagine how much our planet and its organisms have changed through the ages. The Meolingian, which spans from 509 to 490 million years ago, represents a fascinating period of the Cambrian. At this stage, marine biodiversity experiences significant growth, with many groups of animals exploring new environments, developing novel characteristics, and venturing into ecological niches previously unoccupied. Major evolutions in body form and survival strategies are observed, with the emergence of adaptations that will influence the entire animal kingdom for ages to come. The Meolingian is marked by an explosion of biological complexity, sometimes subtle, yet crucial for evolution. The ocean floors, dominated by invertebrates, become the theater of radical innovations in modes of movement, interaction, and protection of organisms. This period marks the emergence of more elaborate predators, equipped with specialized structures, and the consolidation of already well-established groups, like certain arthropods. Marine fauna and flora are in full reorganization, favored by warm climates and a carbon dioxide concentration much higher than today. The sea level, with no polar ice caps, is particularly high, creating vast, shallow zones. However, local variations in salinity and oxygenation sculpt the destiny of biological communities. The Meolingian is divided into three distinct substages: the Wuyuan, the Drumian, and the Guizhongian. Each period unveils significant paleontological discoveries, thanks to key fossil sites and increasingly advanced studies on rock chemistry and fossil formation. These substages reveal the richness and diversity of Cambrian marine life, with fascinating progress in the physiology and behaviors of organisms. The Wuyuan, the first substage of the Meolingian, extends from 509 to 504.5 million years ago. During this interval, Earth experiences a globally warm climate. Shallow seas, known as epicontinental platforms, offer ideal conditions for the proliferation of invertebrates. Coastal habitats, rich in nutrients, allow a diverse range of creatures, from microorganisms to larger animals, to diversify. At this time, the oxygen content in water is uneven across regions, creating contrasting environments. Areas rich in oxygen encourage rapid evolution, while others, poorer in oxygen, hinder the development of complex life forms. Within the Wuyuan, one fossil site stands out: the Burgess Shale, located in the Canadian Rockies and dated to about 508 million years ago. This deposit is one of the most famous in the world for its exceptional preservation of Cambrian fauna. The animals discovered here retain not only their skeletons or shells, but also soft tissues, such as internal organs and muscular parts, offering a unique glimpse into their appearance and lifestyle. This preservation is explained by rapid sedimentation in an oxygen-poor environment, limiting decomposition. A striking feature of the Burgess Shale is the explosion of body plans, with a wide variety of forms suddenly recorded. Some organisms have multiple segments, others have articulated spines, and still others have modified legs for swimming or burrowing. For example, Nectocaris, resembling a small cephalopod, had a primitive jet propulsion system, using its body cavity to expel water and move rapidly, similar to modern squids, though this comparison should be made cautiously. Morella, nicknamed the strangest creature of the Burgess Shale, possesses horn-like cephalic appendages and several pairs of locomotor and respiratory appendages, testifying to the complex organization of an arthropod of the time. This array of body forms illustrates the strong pressure of natural selection during the Cambrian. Each innovation, whether in predation modes, locomotion, or protection against predators, opens up new possibilities for survival or resource exploitation. It is in this context that adaptive radiation emerges, where a group of organisms exploiting a rich environment can split into multiple specialized lineages. The example of the Burgess Shale clearly shows how life, having acquired a flexible anatomical foundation, can produce unusual morphological diversity in a short geological time frame. After the Wuyuan, the history of life enters the Drumian, from 504.5 million to 500.5 million years ago. The Earth continues on a generally warm climatic trajectory, and sea floors become increasingly complex ecologically. This means that in addition to predator-prey interactions, new niches are created, allowing singular life forms to develop. During the Drumian, a remarkable site in the United States, the Margam Formation in Utah, reveals marine fossils dating from this substage and enriches our knowledge of other deposits. In 2024, a discovery causes a sensation. A chordate without a skeleton is perceived as the first confirmed vertebrate outside Asia at this time. Chordates, including fish, amphibians, reptiles, birds, and mammals, are characterized by a dorsal rod, notochord, and pharyngeal slits. Drumian fossils show a specimen without a mineralized skeleton, but exhibiting these distinctive anatomical features. This discovery is fundamental for understanding how the earliest members of the vertebrate group spread geographically. Until then, the oldest chordates close to vertebrates had been primarily documented in China, suggesting that a type of prevertebrate lived in the seas near present-day Asia. Finding an equivalent in North America during the Drumian shows that this group quickly crossed vast oceanic distances, probably thanks to currents or migration cycles that are not yet well understood. It wasn't a conscious journey over thousands of kilometers, but rather a gradual spread, facilitated by environmental changes or the movement of continents, bringing certain land masses closer together. In 2025, a study highlighted a negative excursion, marking a global carbon stress on the Yangtze platform. This negative carbon excursion, marked by a drop in the isotope 13 of carbon in sedimentary rocks, suggests a disruption of carbon stocks and ocean chemistry, likely related to local extinctions or ecological rearrangements. This could be due to a sudden release of organic carbon or a change in photosynthesis. For the Drumian, this excursion indicates stress on marine biospheres, possibly due to low oxygen waters or rapid climatic variations. This study underscores the importance of combining paleontology and geochemistry to reconstruct geological history. In sum, the Drumian substage reveals a dynamic balance. New chordates appear on different continents, while the ecosystem undergoes disturbances. Despite this, life continues to expand, adapt, and renew overall. The third and final substage of the Merovingian, the Gujangian, spans the period from 500.5 to 497 million years ago. This period continues the biological innovations begun earlier, while heralding difficulties for some groups that have thrived since the beginning of the Cambrian. The ecological dynamics of these millions of years combined diversification and sometimes discrete crises, preparing for the major restructurings observed at the beginning of the Ordovician period, just after the end of the Cambrian. In China, in the Linyi region, the Linyi Lagerstätte fossil site offers an exceptional window into marine biodiversity during the Guzhangian. The term Lagerstätte refers to a fossil deposit characterized by exceptional preservation of organisms, either through species abundance or the fineness of preserved anatomical details. In Linyi, scientists have observed a remarkable diversification of arthropods, those invertebrates with a segmented and articulated exoskeleton, such as crustaceans, insects, or arachnids. Several species of arthropods here possess articulated spines, suggesting unprecedented defensive or prey-capturing strategies for the time. Among these groups are predators, known as grapplers, equipped with very developed pairs of anterior legs capable of grasping other organisms. Before this discovery, Cambrian arthropods were mostly seen as scavengers, sifting through the mud or grazers of microorganisms. The emergence of large grasping predators not only indicates a refinement of the food chain, but also increased competition between prey and predators. This evolutionary escalation is reflected in the multiplication of defensive and offensive structures. Moreover, the Guzhangian is marked by a key event in trilobite history. With their segmented shells and compound eyes, trilobites are among the most famous examples of Cambrian fauna. They were successful from the start of the period, occupying various niches and branching into numerous families. However, the Guzhangian records the first trilobite extinction event, known as the Ptychagnostid biomere. The term biomere refers to a rapid and selective extinction, primarily affecting the ptychagnostids. The causes of this local extinction are debated, but sudden environmental changes, such as oxygen fluctuations or disturbances in the carbon cycle, could be responsible. The extinction of these trilobites attests to the fragility of life in the face of ecological variations. Even well-established organisms can see their population decline when environmental conditions change rapidly or extremely. However, it is important to note that, despite these crises, not all trilobites disappeared. Many lineages of other groups persisted, illustrating a recurring phenomenon in the history of life. Localized or moderate extinctions can offer opportunities to other groups capable of adapting to the new conditions created by the disappearance of competitors. Overall, the Merovingian period reflects the rise of increasingly sophisticated marine biodiversity, the multiplication of skeleton and carapace types, the development of complex predatory and defense strategies, and the exploration of new means of locomotion. Animals were not limited to shallow waters. Some descended into deeper areas to find food and shelter, adapting to varied oxygen levels. This development occurred in a planetary context where the climate remained warm and relatively stable despite some fluctuations, and where the high concentration of carbon dioxide favored intense primary productivity in certain regions, thus supporting the food chain. In the Wuyuan, emphasis is placed on the diversity of body plans and the discovery of remarkable forms. In the Drumian, we observe the geographical conquest of primitive chordates, which now appear in North America, in addition to Asia, and environmental imbalances such as the negative carbon excursion in the Yangtze platform. Finally, the Guzhangian tells a contrasting story, with the diversification of arthropods and the first notable extinction of certain trilobite lineages. This sequence of events shows complex marine life, alternating phases of expansion and targeted crises. These successive developments had a long-term impact. The innovations of the Meolingian, such as articulated spines, active predation modes, and the first confirmed forms of chordates on multiple continents, had a lasting impact. They laid the foundations, enabling some groups to endure through ages and evolve into more sophisticated forms. By the end of the Cambrian, many anatomical traits were already in place, preparing the arrival of jawed fish, refined mollusks, and invertebrates with increasingly robust defense structures. Today, we can reconstruct the peculiarities and major trends of the Meolingian thanks to recent methodological advances, particularly between 2023 and 2025. Fossils from deposits are studied using three-dimensional synchrotron tomography, a technique that allows scientists to visualize the inside of fossils without destroying them. This imaging enables the analysis of the arrangement of internal organs, the structure of appendages, and the shape of soft tissues. Meanwhile, Raman spectroscopy performed directly on fossils provides information about their chemical composition, even revealing the presence of fossil eye pigments. This helps answer long speculative questions about the visual capacity of Cambrian species. This approach also allows for the detection of organic residues, providing insights into their diet or the structure of their cuticular envelopes. In terms of geological dating, the ultra-high-resolution UPB method, with uncertainties of less than 1/10 of a million years, solidifies the chronology of these events. It is often applied to volcanic ash trapped in sediments, allowing each discovery to be precisely anchored on the geological timescale. Coupled with astrochronology, which studies astronomical cycles, particularly those related to Milankovitch cycles, this method has refined the chronological understanding of the Meolingian. Subsequently, the marine world will continue its journey toward new revolutions, crossing the Cambrian-Ordovician boundary to experience new expansions and evolutionary crises. But the foundations observed during the Meolingian lay the groundwork for the future complexification of ecosystems. The anatomical and ecological achievements, from the primitive chordates of Utah to the grappler arthropods of Ligny, testify to how the tree of life branched out at this time. By following the traces of these creatures and analyzing their fossilized environments, scientists continue to unravel the evolutionary puzzle that shaped the biosphere as we know it. Studying the Meolingian is to become aware of the speed and scope of biological changes on Earth. In just a few million years, multiple innovations appear. Some species thrive, others decline, and the marine world is profoundly transformed. Observation of these ancient stages, illustrated by geological formations, informs us about the history of life and its ability to adapt to planetary upheavals. This in-depth diversification, despite its localized crises and extinctions, has greatly contributed to the richness of current life on Earth. The Furongian, which spans from 497 to 485.4 million years ago, marks the end of the Cambrian. This period is characterized by major changes in marine life and the biosphere, with biological and environmental events profoundly transforming ecosystems. The Furongian is divided into three substages: the Paebian, the Zhongshannian, and Stage 10. Each stage presents distinct characteristics, such as the diversification of animals, trilobite crises, and geochemical indicators of planetary transformations. We will explore the evolution of fauna, flora, marine environments, and ecological crises during this key period. The Paebian, from 497 to 494 million years ago, corresponds to the first part of the Furongian and is marked by a major geochemical event, the Spice Event. Spice stands for secular positive carbon isotope excursion, or a positive delta-13 carbon excursion. Delta-13 carbon measures the proportion of carbon-13 isotope relative to carbon-12 isotope. This measure allows the detection of changes in the global carbon cycle. During the Spice event, a rise in delta-13 carbon is observed, often correlated with an enrichment in oxygen in some shallow oceanic regions, such as tropical platforms. Why is a carbon excursion important? Because variations in the carbon cycle are often linked to major planetary changes affecting global temperature, oxygen distribution in the ocean, or the proliferation of biological communities like algae or bacteria. Oxygen enrichment is linked to these upheavals, as they lead to changes in the composition of the atmosphere and marine waters. With more oxygen, marine ecosystems can see the emergence of new ecological niches, favoring the diversification of megafauna, especially animals with mineralized skeletons. Lepibien highlights the geographical spread and abundance of certain groups of marine invertebrates, notably the lingualiform brachiopods. These bivalve-shelled animals, similar to today's bivalve mollusks, have different internal anatomy. Their body is organized in such a way that it attaches to the marine substrate through a fleshy stalk. During the Pabian, these brachiopods became widespread in different marine environments, and their diversity increased, reflecting an evolutionary success at the end of the Cambrian. Beside the brachiopods, another group draws attention: the phosphatic test foraminifera. These unicellular protozoans are known today primarily in a calcium carbonate-covered form, as seen in many modern planktonic foraminifera. However, during the Paebian, some possessed a phosphate test. This phenomenon likely occurs in environments where phosphorus availability and water acidification differ from those observed today. Discoveries of these phosphatic test foraminifera in Paebian sediments revealed diverse mineralization strategies in Cambrian plankton and a subtle geochemical balance. Moreover, the climate of the Paebian remains generally warm. Although we lack direct temperature measurements for this period, paleoclimatic reconstructions suggest values higher than today, with a globe without permanent polar ice caps. Some estimates indicate surface waters often exceeding 20 degrees Celsius in tropical and subtropical regions. Such a thermal context would favor the rapid proliferation of marine life, reinforced by the oxygen increase detected during the Spice event. After the Paebian, we enter the Zhongxianian, which extends from 494-489.5 million years ago. The atmosphere and oceans remain warm and rich in carbon dioxide, but local variations in ocean circulation and oxygenation begin to manifest. A significant event of this stage is the appearance of vertical bilaterian burrows, indicating that some animals, presumably burrowing organisms, began to exploit the sediment column, penetrating more than 10 centimeters below the marine seabed. To understand this advancement, it is important to remember that at the beginning of the Cambrian, burrowing behaviors were shallow and horizontal. Organisms could not venture deeply into the substrates due to a lack of oxygen, anatomical limitations, or other ecological factors such as surface predation. The discovery of vertical burrows more than 10 centimeters deep before the end of the Cambrian shows that certain animal groups crossed an important threshold in terms of locomotion, respiration, and adaptation to the environment. This deeper bioturbation altered the structure of the sediments, improving aeration and nutrient circulation. These geological footprints attest to the rise of benthic ecosystem complexity. Alongside this innovation in bioturbation, the Jiangshanian is also associated with a faunal crisis episode, the Sepkoski trilobite III. Jack Sepkoski, a paleontologist, identified several phases of trilobite extinction. The Sepkoski trilobite III crisis marks a massive decline in the red lacide trilobites, primitive trilobites, common at the beginning of the Cambrian. Their gradual disappearance reflects ecosystem transformations and the increasing competition with other arthropods more efficient in feeding and mobility. These extinctions seem to coincide with sea level fluctuations and changes in ocean chemistry, although determining the exact causes remains complex. It is possible that a local oxygen drop in some basins, combined with climatic variations and competition with new groups, accelerated the demise of these trilobites. Fossils found in China, Morocco, and North America show the fragility of biological assemblages of the time. However, other trilobite groups, better adapted or colonizing specific niches, managed to maintain their presence. Alongside these crises, the Jiangshanian sees the persistence of brachiopods, mollusks, and phosphatic-shelled foraminifera in various coastal and subcoastal regions. Climatic conditions continue to favor ecosystems in warm seas, but the sometimes more unstable dynamics of oxygenation already herald the end of the Cambrian. Scientists envision oceans with complex circulation, with deep oxygen pore zones, forming barriers for certain groups. The study of sulfur and nitrogen isotopes in these sediments hints at cyclic modifications of upwelling systems, sometimes rich in nutrients but depleted in oxygen, creating euxinic conditions. This forces underwater communities to adapt or move rapidly. The last segment of the Furongian, provisionally called Stage 10, covers the period from 499.5 million to 485.4 million years ago. Although this stage has not yet been officially named, several scientists debate the relevance of establishing a global boundary, stratotype section and point, GSSP, in different regions. Two main sites are often mentioned, near Dwibian in China and Lawson Cove in Utah, USA. A GSSP is a geological outcrop that clearly shows the chronological boundary between two subdivisions of geological time. This discussion underscores the importance of international scientific issues. Assigning a name and reference point facilitates global comparison of stratigraphic layers. The 10th stage is particularly marked by the transition between the end of the Cambrian and the onset of the Ordovician. During this period, marine fauna continues to evolve, but some major Cambrian lineages give way to groups destined to thrive during the Ordovician. Eopatognathus conodonts, small marine organisms with microscopic dental elements, are among the most useful fossils for geologists. Sensitive to oceanic chemical changes, they appear and disappear over short periods. The species Eopatognathus is particularly associated with a major oceanic change between the Cambrian and Ordovician, signaling a reorganization of fauna and a disruption in the chemical balance of the seas. During this period, a reconfiguration of ocean currents is suspected, influencing the distribution of oxygen at higher latitudes. The Earth, fragmented into isolated paleocontinents, experienced mild or temperate warm climates with coastal configurations favorable to sparse reefs. These reefs, less dominated by archaeocyaths, are now supported by siliceous sponges and bacterial biofilms, compensating for the decline of reef builders. However, the rise of algal blooms and the proliferation of phosphatic-shelled microorganisms can slow rapid carbonate formation, making marine landscapes more heterogeneous. The coupling between climate, sea level, and ocean chemistry is essential for understanding the transition to the Ordovician. When marine waters warm or change composition, they impose new challenges on animals: adaptation, migration, or extinction. Trilobites, increasingly threatened by successive crises, continue to exist in more resilient forms. Concurrently, emerging groups of cephalopods, brachiopods, and graptolites, often planktonic colonial animals, gradually establish themselves. Scientists examine the last stage of the Cambrian to identify signs of this biological renewal that will mark the Ordovician: a sustained complexification, the appearance of more elaborate reefs, and new superpredators. By assembling the three key moments of the Furongian Paibian, Jiangshanian, and Stage 10, we obtain a dynamic scenario where the Cambrian biosphere confronts a transitioning environment. Chronologies show that biological evolution, ocean chemistry, and climatic conditions are intimately linked. For example, the increase in delta-13 carbon during the Spice event fits into a context of increased oxygenation of marine platforms, favoring the diversification of certain invertebrate lineages. Linguliform brachiopods and phosphatic-shelled foraminifera illustrate the complexification of mineralization, while the first deep bioturbation demonstrates the ability of certain organisms to profoundly transform their environment. Simultaneously, we also witness occasional crises, like the Sepkoski Trilobite III crisis, which hasten the erosion of certain dominant lineages, giving way to groups better equipped to survive environmental changes. The concept of dynamic biodiversity is fully realized here. Life does not stagnate. It adapts, redistributes, and sometimes vanishes in the face of environmental modifications. This phenomenon of partial resetting of fauna is reflected in all major biological crises throughout time, and the Cambrian, though an era of diversity explosion, is no exception to this rule. The Cambrian closes, and the transition to the Ordovician crystallizes in the geological layers of Stage 10. The conodonts Iapetognathus mark a significant milestone for distinguishing the boundary between these two systems, revealing possible redistributions in ocean circulation and suggesting a profound faunal renewal. Discussions around the position of the GSSP for this stage illustrate the importance of fieldwork and the effort of international stratigraphic correlation in geology. To understand the Furongian, let's describe the general environment of the time. The continents do not resemble our current Earth. Most of the land masses are located in the southern hemisphere, with Gondwana, including South America, Africa, Australia, Antarctica, and India, but in different configurations. Other land masses like Laurentia and Baltica move at various latitudes. This division of the lands directly influences the position of epicontinental seas and sea level, key factors in biodiversity distribution. On the continents and along coastal areas, the flora is rudimentary. There are no complex terrestrial plants yet. Vegetation is limited to algae and bacterial biofilms present along shores or in aquatic environments. Large reefs, dominated by cyanobacteria or siliceous sponges, succeed the archaeocyathids, reef builders at the beginning of the Cambrian, which declined before the Furongian. Although algae fossilize poorly, they play a role in converting carbon dioxide into oxygen and biomass, thus fueling the marine food chain. In the waters, the fauna comprises a wide range of invertebrates: trilobites with articulated exoskeletons, stationary brachiopods, various mollusks, primitive echinoderms, and a series of non-trilobite arthropods, such as the first crustaceans or ancestors of shell-filtering animals. Marine predators include radiodonts, close to Anomalocaris. These are formidable jawed worms, and possibly chordates already practicing visual hunting. During the Furongian, some predators increase in size, and their mouthparts diversify, indicating the strong selective pressure they exert on their prey. All these new organisms and behaviors testify to an intensification of trophic interactions. Evolutionarily, this dynamic sets the stage for the Ordovician, where marine biodiversity will take a leap forward. By the end of the Furongian, we can already perceive the beginnings of a great expansion of life, marked by the evolution of increasingly complex organisms. Besides its descriptive interest, the Furongian offers fundamental lessons that fuel contemporary research. Understanding how variations in the carbon cycle are linked to oxygenation events, providing a model to analyze past crises and anticipate the impact of modern changes on climate and biodiversity. Measuring how bioturbation of the seafloor alters nutrient cycles, with burrows and galleries trapping or releasing chemical substances, thus influencing ecosystem functioning. Analyzing the dynamics of partial extinctions, like the Sepkoski trilobite III crisis, showing how a flourishing group can disappear in favor of better adapted forms. Developing precise dating methods to delineate stratigraphic boundaries, like the issue of the GSSP for the 10th stage, illustrating the complexity of chronological limits essential to global correlations. At the end of stage 10, nature is preparing to embark on a new phase of marine radiation during the Ordovician. Eoptognathus conodonts serve as stratigraphic markers. They indicate that the Cambrian is giving way to a renewed world where brachiopods and cephalopods, among others, will rise. With the decline of certain trilobite lineages and the increase in ecological niches, the stage is set for future biological innovations. The Furongian shows that the history of life is made of cycles. Crises punctuate this complex evolutionary story, catalyzing the emergence of more sophisticated organisms. For the current scientific community, studying this period helps to understand the mechanisms of biodiversity and the resilience of species in the face of climatic, geological, and ecological challenges. Thus, from the Cambrian to the Ordovician, we leave a pioneering era to enter another, where the advances and upheavals of marine life will continue to shape the Earth. At the beginning of the Cambrian, new forms of life and novel anatomical traits appear, durably altering the trajectory of life on the planet. If we could immerse ourselves in the vast Cambrian seas, we would witness an effervescence of creatures with morphologies, sensory organs, and lifestyles radically different from anything that existed before. This narrative focuses on the internal innovations of the Cambrian, such as sensory systems, musculature, internal skeletons, arthropod cuticular ribs, and emerging trophic networks that structure predation and food distribution. When examining the dawn of animal life, one of the most spectacular changes is the emergence and refinement of sensory organs. Before the Cambrian, living forms remained very simple, with limited perceptual abilities. But entering this period, there are signs of a real leap forward in how animals perceive and interact with their environment. The best example of this sensory revolution is undoubtedly the appearance of true compound eyes. The term compound eyes refers to structures formed of multiple facets, each connected to a photoreceptor cell. Imagine a visual mosaic offering a fragmented but very wide view, sensitive to movements. In a Cambrian creature named Alalcomeneus, fossils reveal eyes so precise they could distinguish details as small as a millimeter. Each facet, acting as a light sensor, equates to a resolution capable of detecting fine variations in this space. Such a system allowed the detection of prey or predators at close range, offering a significant evolutionary advantage. In addition to compound eyes, many Cambrian animals possessed long facial appendages, similar to antennas, which help them perceive their environment. These organs, known as chemomechanosensitive, combined chemical detection such as smelling or tasting compounds in the water and the perception of movements, such as vibrations or currents. This allowed the animal to literally sniff and feel its surroundings. In seas where food competition intensified, detecting a slight current or a chemical trace offered a valuable advantage. To place these innovations in the broader context of the Cambrian, imagine these growing sensory capacities encouraging an arms race in which prey develop strategies for camouflage or escape, while predators further perfect their senses and weapons. Eyes improve, antennas diversify, and communication between individuals becomes more complex, creating, over millions of years, chains of interactions infinitely richer than in earlier epochs. In parallel with this sensory revolution, another major change occurs. Some animals develop more powerful musculature, and in a few, the beginnings of an internal skeleton appear. While external skeletons like the rigid cuticle of arthropods are common, the concept of an internal supporting structure remains exceptional at the start of the Cambrian. Yet, in a creature named Pikaia, this innovation is observed associated with V-shaped chevron musculature, evoking the letter V. These muscle segments, called V-shaped myomeres, mark a fundamental advancement in body organization. Pikaia appears as a frail organism, resembling a flattened ribbon a few centimeters long. Considered a basal chordate, a probable ancestor of vertebrates, it presents V-shaped myomeres, very similar to those later observed in primitive fish. These segmented muscles, distributed all along the body, offer particularly efficient contraction thanks to their shape. Imagine an accordion whose every fold contracts rhythmically. This structure allows the animal to move with flexibility and speed in its aquatic environment. In Pikaia, the internal skeleton remains rudimentary, but represents a crucial step. Unlike an external skeleton, which protects the body's surface, an internal structure supports the entire organism and allows muscles to exert a more targeted force, useful for swimming or crawling. Although Pikaia is not a formidable predator, this innovation lays the groundwork for the emergence of fish, and then vertebrates. These myomeres already herald future diversity. Without this anatomical organization, fish, amphibians, reptiles, birds, or mammals would never have existed in their current form. While the chordates begin to develop an internal skeleton, others rely on a sophisticated external envelope. The arthropods, in particular, experience a remarkable boom in the Cambrian. Although they appeared earlier, it is during this period that their anatomy stabilizes and perfects itself. Their rigid cuticle, an external shell they must sometimes molt to grow, incorporates cuticular ridges, sorts of reinforcements that ensure protection and rigidity while maintaining a certain flexibility at the joints, essential for movement. A joint, in biology, is an area where two parts of the body can move relative to one another. In arthropods, the body is segmented in a modular way, and each limb is divided into several articulated articles. The biramous limbs have two branches, one dedicated to locomotion, walking or swimming, and the other to respiration or food filtering. This versatility allows arthropods to occupy very diverse environments simply by adapting the function of their appendages as needed. During the Cambrian, segmented structures and numerous joints offer great precision in movement. An arthropod can bend a segment or deploy a claw to grasp prey, all while remaining stable. The rigid cuticle, reinforced by ridges, also effectively protects against predators. This advancement is crucial in a competitive environment. Thanks to this natural engineering, these animals move like small biological robots, exploring the ocean floor, digging burrows, or filtering food carried by currents. Even if segmentation exists in several animal groups, the Cambrian arthropods have pushed it to a high degree of specialization. Each segment carries a pair of appendages with distinct functions, legs for walking, sensory palps, or gill extensions. This organization already foreshadowed the diversity of modern crustaceans, arachnids, and insects, who inherit this ability to adapt their limbs to specific tasks, essential to their evolutionary success. These anatomical evolutions, vision, locomotion, and support, make perfect sense when placed in the ecosystem. It is during the Cambrian that the first complete trophic networks are formed, that is to say, the food chains connecting producers, grazers, and predators. To understand this dynamic, let's first focus on the essential actors of life, the primary producers, at the base of every food chain. In the oceans, primary producers are mainly microscopic algae and photosynthetic bacteria. They are also found as biofilms, thin layers of microorganisms that cover surfaces, capturing light to produce their energy. These organisms form the base of almost every marine food network. If the biosphere were a building, they would be the foundation. Without them, no food chain could exist, as they create the food necessary for all other living beings. Grazers follow the primary producers by consuming these microorganisms. Among them, trilobites with segmented shells often adorned with spikes or ridges. They moved on the seabed, scraping biofilms and microscopic algae for food. Their great diversification is due to their ability to occupy various niches. Some dug into benthic deposits, others captured tiny organisms suspended in the water, thus ensuring a constant food supply. At the top of the food chain are predators, notably radiodonts, the most famous being Anomalocaris. These animals impress with their size, often several tens of centimeters, or even more than a meter, and their anterior appendages shaped like pincers to seize their prey. Their mouth, a disc equipped with denticulated plates arranged in a ring, functions like a muscular funnel. A careless prey caught in these circular jaws stands little chance of escaping. The cascading trophic network relies on solar energy converted into biomass by algae and biofilms. Grazers consume this biomass, while superpredators hunt grazers or other marine animals. This system organizes food relations, regulates populations, and stimulates evolution, as each species must adapt to survive. It also influences the chemistry of the water, since each level releases wastes and recycles essential nutrients for the ecosystem. The biological innovations of the Cambrian work in chain. The compound eyes enhance camouflage and escape, while the V-segmented muscles enable active locomotion and the exploration of varied habitats. Reinforced cuticles provide protection and stability, helping to conquer new niches. Meanwhile, the complexity of trophic networks intensifies competition, pushing each species to constantly adapt by refining their senses or developing stronger armor to survive in this changing world. This evolutionary process acts like a red thread in the Cambrian. Innovations adapt to environmental pressures and interactions between species. Better sensory perception stimulates hunting, which in turn leads to the development of defenses, creating a continuous circle. In this game, the slightest advantage can determine survival or extinction. The Cambrian thus illustrates this fundamental principle. The success of a lineage depends on its ability to innovate and take advantage of ever-changing ecological conditions. To a modern observer, it seems incredible to deduce the presence of sensory organs, muscles, or an internal skeleton from often fragmentary fossils. Yet, paleontology has recently benefited from new techniques, allowing much further advances than a century ago. For example, the mention of cuticular ridges in a Cambrian arthropod comes from visible lines on its fossilized shell. Similarly, by meticulously studying fossil appendages and comparing them to current species, one can deduce the probable functions of chemomechanosensitive antennae. The discovery of fossils in Pikaia showing the famous V-shaped myomeres has deeply marked specialists, confirming in these early chordates a muscular organization close to that of fish. Each fragment of Cambrian rock reveals a secret: a shell contour, an articulated limb, an eye remnant. Thanks to advanced tomography, unknown details, like fossilized nerve fibers, can also be spotted. In addition, the chemical study of isotopes allows precise dating of these layers and tracing the chronology of biological innovations. When one measures the extent of these advances, from refined senses to segmented muscles, from the internal skeleton to articulated cuticles and the complexity of trophic networks, one realizes the journey made since the first multicellular organisms. The success of current arthropods, present on land and in sea, is largely explained by this body modularity, which allows them to specialize their limbs and molt their shell to grow. These innovations were the key to their initial and lasting success. In chordates, the V-shaped myomeres of Pikaia herald the future diversity of vertebrates, fish, amphibians, reptiles, birds, and mammals. Without this muscular and structural foundation, the spine and complex musculature we know would not have emerged. Simultaneously, sensory systems evolved toward ultra-sophisticated eyes, whether in insects, cephalopods, or vertebrates. The Cambrian thus marks the dawn of an evolutionary explosion, laying the foundations of the great current lineages of life. These sensory and anatomical innovations are inseparable from the ecological interactions that nurtured them. For example, developing a compound eye requires a significant energy investment, justified only if it provides an advantage, such as avoiding a predator or hunting more effectively. Thus, the sophistication of the senses accompanies the intensification of the arms race. The more effective predators become, the more prey must improve their defenses, driving ever more complex adaptations in a continuous evolutionary cycle. The trophic cascade closely links each link. The availability of algae limits the grazers, which in turn condition the super predators. When all goes well, these energy flows favor the diversification of trilobites, arthropods, and other invertebrates. But a disruption, such as a chemical change in the water or a decrease in photosynthesis, can weaken the entire chain. Thus, the biological innovations of the Cambrian are not just isolated feats. They testify to a global ecological dynamic, weaving links between all forms of life. Today, modern fauna bears the legacy of the Cambrian. The compound eyes, varied in insects and crustaceans, testify to this sensory evolution. The V-shaped musculature of fish descends directly from forms like Pikaia. Terrestrial arthropods, with their inherited modular segmentation, have adapted to almost every environment. Finally, the rudimentary internal skeleton observed in some animals announces the formidable rise of vertebrates, of which we are a part. this distant past still shapes current life. The first forms of these innovations sometimes disappeared with their carriers, victims of the many crises and extinctions in Earth's history. Only a few distant descendants persist among the Cambrian arthropods. Yet, these innovations opened evolutionary pathways with countless ramifications, traces of which we still observe today. The Cambrian perfectly illustrates experimental evolution, a phase of intense biological exploration where some forms thrived while others vanished. The Cambrian reveals a golden age of marine life, where evolutionary ingenuity is fully expressed. The perfected sensory systems give animals a new awareness, enhancing hunting and fleeing. The segmented musculature and a primitive internal skeleton mark the beginnings of chordates, ancestors of vertebrates. In arthropods, the reinforced cuticle and sophisticated joints transform defense, locomotion, and adaptation to environments. Finally, cascading trophic networks organize energy flow, shaping the feeding and survival strategies of each species. These innovations radically transformed the living world. Rather than remaining passive on muddy grounds or floating in search of particles, many animals adopt active behaviors, exploring, hunting, defending. This increase in activity energizes ecosystems, capable of supporting a large diversity of life forms, from producer microorganisms to imposing top predators. Although these creatures may seem strange to us today, they are the first to bear key innovations. Without this anatomical, sensory, and ecological impetus, animal life would have remained simple. Whether it's the compound eyes of Alalcomaneus, the V-shaped myomeres of Pikaia, the biramous limbs of arthropods, or the great trophic cascade from algae to radiodonts, it was a bustling world where each advancement counted for survival. When observing an insect with compound eyes, the colors in a fish's retina, or the segmentation of a millipede, we pay homage to the experiments of the Cambrian. These innovations have forged the diversity and complexity of living beings we inherit. They explain the widespread vision, articulated modular bodies, and hierarchical food chains, from microorganisms to super predators. The Cambrian period is a time of climatic and geological upheaval, marked by a super greenhouse effect, where carbon dioxide levels were much higher than today. The absence of permanent polar ice caps altered ocean circulation and global climate. In this text, we will explain how these conditions, a high CO2 level, specific marine chemistry, and the upwelling of oxygenated waters, influenced the diversity and extinctions of marine fauna. We will also see how Cambrian ecosystems were structured by variations in oxygen in coastal waters, alternating between favorable and depleted periods. The phenomenon of a powerful greenhouse effect corresponds to the ability of certain gases in the atmosphere, notably carbon dioxide, to trap the heat emitted by the Earth. The higher the concentration of CO2, the more heat remains trapped, causing global warming. In the Cambrian, carbon dioxide levels are believed to have exceeded 10 times those of the pre-industrial era, far beyond the natural variations observed in recent geological periods. This intense warming profoundly influenced the climate and marine living conditions. To imagine Cambrian landscapes, one must envision an Earth almost without permanent ice caps. Unlike today, when Antarctica and Greenland are covered with ice, these regions were then free of enduring ice. This absence raised sea levels, flooding vast portions of the continents under immense shallow seas. These coastal areas, rich in light and nutrients, were essential habitats for marine life. They offered large areas of sedimentation in warm waters, promoting the diversification of organisms. These vast aquatic expanses can be seen as true incubators. The planet was generally warmer than today, with average temperatures possibly exceeding 25 to 30 degrees Celsius, even in temperate or near polar regions. This hot climate intensified exchanges between the atmosphere and the ocean, accelerating the circulation of carbon and nutrients in ecosystems. A positive feedback maintained this cycle. Heat favored the release of carbon dioxide, which in turn sustained warming, a persistent phenomenon over millions of years. Carbon dioxide, largely produced at this time by intense volcanism and the metabolism of early living organisms, influenced not only temperature, but also the ocean's chemistry. As its concentration increased, the ocean absorbed part of it in a process called carbon dioxide dissolution. This dissolution altered the seawater pH by making it more acidic, as CO2 partially transformed into carbonic acid. However, in the Cambrian, the situation was very different from today. Marine species were in their early evolutionary stages, and many had not yet developed complex calcareous structures. In the Cambrian, before the explosion of solid shells, marine life mainly consisted of organisms with minimally mineralized or soft shells. The first rudimentary skeletons in phosphate or carbonate were only just appearing. Therefore, the ocean's chemistry mainly influenced species distribution without causing massive shell dissolution as seen today. This constantly changing environment forced organisms to adapt to variations in pH, chemical composition, and intense competition. To understand the dynamics of this perpetually adapting life, it is essential to emphasize that the Cambrian Ocean was not uniform. Coastal areas, near nutrient sources, continental inputs, runoff, volcanoes, and hydrothermal vents, had conditions that were very different from deeper regions. In shallow areas, heat combined with sunlight created highly productive spaces where microorganisms proliferated in abundance. These microorganisms, whether photosynthetic bacteria or algae, promoted the formation of biofilms, real biological mats, adhering to marine sediments, and serving as habitats for other life forms. Beyond the key role of carbon dioxide, variations in dioxygen have also influenced the evolution of fauna. According to a 2024 study published in Nature Geoscience, a slight surplus of oxygen, particularly in coastal or favorable zones, triggers significant biological diversification. This gas, essential for animal respiration, encourages the development of complex creatures near benthic and planktonic communities. This allows for high metabolisms, active predation strategies, and a greater diversity of species. During the Cambrian, local variations in dioxygen came from several mechanisms. Marine photosynthesis, especially by planktonic algae and certain bacteria, released dioxygen into the water. Ocean circulation then redistributed this oxygen, bringing it from illuminated surfaces or consuming it in areas rich in decomposing organic matter. These small fluctuations influenced local faunal diversification, as species capable of benefiting from a slight oxygen surplus gained an evolutionary advantage over those living in poor environments. Even though the global amount of dioxygen during the Cambrian was lower than today, the ecosystems of the time did not require the same levels to thrive. Life had just crossed key milestones, and organisms reacted strongly to the slightest increase in oxygen. These minor local surpluses, confirmed by geochemical analyses, were enough to favor the emergence of more complex animals and the first structured food chains. A key aspect of the Cambrian climate was ocean circulation, these vast currents that redistributed heat, nutrients, deoxygen, and mineral salts. At that time, active plate tectonics placed continents in very different configurations, some grouped together, others isolated in microcontinents. This fragmentation favored the emergence of shallow epicontinental seas, where currents reacted to temperature and salinity variations. We call Euxinea marine zones, with very little dioxygen and often enriched with sulfur compounds. There, decomposition consumes almost all the oxygen, creating anoxic deep layers where bacteria produce hydrogen sulfide, toxic to most animals. In the Cambrian, these areas caused mortality in poorly adapted species. Those able to flee these toxic waters had an evolutionary advantage. Fossils sometimes show local extinctions linked to these anoxic and sulfuric phases. The upwelling of oxygen-rich waters, often from the surface to deeper areas, revitalized certain ecosystems and allowed the establishment of new communities. This alternation between oxygen-poor and oxygen-rich periods created waves of diversification and extinction. In a few thousand or million years, fauna drastically renewed itself in a region. These fluctuations, immense on our scale but brief for geology, make the Cambrian an era of rapid biological experimentation. To understand variations in ocean circulation, several factors must be considered. The absence of polar ice caps altered the thermal gradient between the equator and the poles. Usually, polar ice produces cold, dense waters that plunge deep, feeding the major currents. In the Cambrian, with warmer poles, this thermohaline circulation, the large loop that mixes surface and deep waters, functioned differently, sometimes less stably. Another key factor came from the Earth's high internal heat, due to sometimes intense volcanism. Submarine or continental eruptions released carbon dioxide and nutrients mixed with ash and gases, locally altering water composition. Combined with tectonic movements, these variations led to rapid sea level changes. Rising and falling modified sedimentation and the shape of basins, influencing currents and stratification or, conversely, vertical mixing of waters. These climato-tectonic interactions made the Cambrian Ocean very heterogeneous. At any given time, some basins had clear, oxygen-rich waters, while other zones, like bays or continental slopes, plunged into anoxia. Adaptable organisms, able to quickly change environments or tolerate variable conditions, fared better than those with narrow ecological niches. Fossils show a great diversity of adaptations, such as the ability to slow metabolism to survive in oxygen-poor environments. In the carbon dioxide-rich atmosphere of the Cambrian, the high temperature, strong coastal productivity, and diversity of depths created a favorable environment for evolution. The major top predators had not yet been established. Ecosystems were being built as fauna developed new techniques for hunting, movement, and defense. In this context, protists, soft-bodied animals, and future arthropods were able to expand, explore new niches, and exchange genes and morphological innovations. Variations in dioxygen favored the appearance of creatures with efficient respiratory systems capable of exploiting these conditions. Active predators took advantage of local oxygen enrichment episodes to support a fast metabolism, powerful muscles, precise coordination, and enhanced sensory capabilities. This evolutionary race, linked to fleeing or searching for better oxygenated waters, stimulated the emergence of robust anatomies, segmented, sometimes reinforced with exoskeletons. On illuminated marine plateaus rich in nutrients, photosynthesis produced a large amount of organic matter, supporting the multiplication of herbivores and the formation of complex food chains. Dead organic matter accumulated at the bottom, enriching the sediments, but could locally deplete oxygen, thus favoring anoxic or euxinic conditions. Climate influences not only temperature, but also primary production, water circulation, and an ecosystem's ability to renew itself and maintain its biodiversity. The Cambrian is famous for the sudden emergence of complex life forms, often called the explosion of life. However, this period also experienced phases of stagnation or decline in diversity. Paleontological and geochemical studies suggest that these declines sometimes coincide with the expansion of anoxic oceanic zones or temperature changes. The warming of the waters, their acidification, and the scarcity of dioxygen contributed to local, even regional extinctions of more fragile species. When the balance was restored, for example, thanks to a change in ocean circulation bringing oxygenated water, diversification resumed. This broadened the range of food resources, creating new ecological niches for opportunistic organisms. This succession of extinction and diversification phases shows the great sensitivity of Cambrian ecosystems to environmental factors. A small chemical variation, like an increase in dioxygen, was sometimes enough to trigger a cascade of transformations and the emergence of innovative species. Mass extinctions, like those at the end of the Permian or the Cretaceous, are not systematically linked to the Cambrian. However, the periodicity of boom and bust episodes is well documented in certain sedimentary basins. Several explanations are proposed, such as orbital cycles, volcanic activity, and the reconfiguration of continental blocks. But it is above all the fragile balance between carbon dioxide, dioxygen, nutrients, temperature, and ocean circulation that determines the conditions of these pulsations. To uncover the secrets of Cambrian environments, researchers rely on geochemical and geological analysis techniques. Although there are no direct observations of this ancient atmosphere, rocks preserve valuable clues: carbon and sulfur isotopes, traces of metals, microscopic fossils of plankton or bacteria, and sometimes even fossilized pigments. Marine sediments, especially those preserved in anoxic environments, form remarkably stable archives of ocean chemistry at the time of their deposition. By determining the levels of dioxygen or carbon dioxide trapped in rocks, scientists reconstruct the variations that affected the global ocean. By cross-referencing these data with fossils found in the same layers, they establish a link between ocean chemistry and biodiversity evolution. Recent research highlights the great sensitivity of Cambrian ecosystems. Even small increases in dioxygen can lead to significant biological upheavals by altering respiration, metabolism, or food chains. Limiting ourselves to the climate of the Cambrian, we see how this era served as an evolutionary laboratory. A super greenhouse effect imposed unstable conditions, accelerated composting of organic matter, chemical upheavals, dead zones, and oxygen-rich oases. Organisms capable of migrating, innovating physiologically, or surviving in oxygen-poor environments laid the foundations for particularly adaptable lineages. The absence of polar ice caps recalls certain warm phases of the Mesozoic or Cenozoic, characterized by high sea levels. But in the Cambrian, a still very primitive fauna experimented for the first time with major innovations: shells, exoskeletons, burrowing strategies. In this context, carbon dioxide and ocean circulation played a catalytic role, accelerating the emergence of adaptations exploiting these changing environments. Today, understanding how Cambrian ecosystems reacted to climate and chemical fluctuations helps anticipate the responses of modern environments to current disruptions. Although Earth has undergone profound biological evolution, the logic remains similar. An excess of carbon dioxide warms the planet and disrupts oxygen cycles. Marine, coastal, or open environments then become zones of adaptation or extinction, depending on the availability of oxygen, a vital element for many species. In summary, several essential traits characterize the climate and environments of the Cambrian. Super greenhouse effect, very high levels of carbon dioxide, leading to marked global warming. Absence of permanent polar ice caps, high sea level, shallow seas, low thermal gradient disrupting ocean circulation. Variable oxygen fluxes, slight local enrichments were enough to trigger biodiversity booms. Unstable ocean circulation. Absence of ice and active tectonics sometimes favored stagnation of oxygen, poor deep waters, or oxygenated upwellings, conducive to diversification. Rapidly evolving ecosystems. These extreme conditions constantly stimulated the emergence of new niches and biological strategies. Thus, the foundations of a more complex biosphere were laid, paving the way for major evolutionary radiations. Understanding this dynamic shows how the Cambrian Earth was already a living world, saturated with carbon dioxide and animated by a changing ocean circulation, conducive to the emergence of unprecedented biodiversity. This warm climate and oxygen variations allowed the appearance of the first sophisticated interactions between predators and prey, as well as structured communities. The Cambrian is the cradle of complex life, as its extreme conditions favored unprecedented biological innovation. When we talk about Lagerstätten, we refer to exceptional fossil deposits, where the fine preservation of organisms offers a unique window into ancient life. Here, we explore the Great Lagerstätten of the Cambrian, a period of spectacular rise in animal life. These sites allow us to reconstruct the appearance, lifestyle, and environment of extinct creatures, often very different from those we know. From Chengjiang, 518 million years ago, to Kaili, 497 million years ago, follow us through 7 key Lagerstätten to discover surprising ecosystems, major biological innovations, and the first foundations of a burgeoning biodiversity. Let's start with Yunnan Province in southwest China, where the extraordinary Chengjiang Lagerstätte is located, dated to about 518 million years ago. This site of the Lower Cambrian reveals remarkable marine diversity. Before its discovery, our knowledge was mainly based on fragmentary fossil traces. Here, exceptional conservation allows us to observe not only hard parts, like shells, but also sometimes soft tissues, gills, digestive system, even muscles. Chengjiang geological formation was deposited at the bottom of a shallow sea, where frequent deposits of fine sediments quickly buried dead animals, protecting them from decomposition and scavengers. This rapid accumulation, combined with a low oxygen level, allowed for exceptional fossilization. At that time, the Earth was experiencing warming with a high concentration of carbon dioxide. The coasts of Yunnan were bathed in warm waters, around 20 to 30 degrees C, fostering marine biodiversity far richer than imagined for this early Cambrian era. Chengjiang owes its fame to the preservation of a wide variety of species, belonging to extinct groups or to lineages still present today. There we discover the first arthropods, ancestors of insects, crustaceans, and arachnids, as well as creatures endowed with a notochord, an early sign of chordates. Some fossils even reveal primitive sensory organs, less complex than today, but already adapted to evolve in a world of burgeoning biological expansion. This incredible diversity of animals, with varied body forms, illustrates what is called the initial explosion of body plans. It is not an explosion in the literal sense, but a period of rapid evolution where new body types appear in a short time. Paleontologists continue their research on this site to better understand how life crossed this crucial threshold, a pivotal moment still shrouded in mystery. Thanks to its remarkable preservation, Chengjiang remains the first stage of our exploration of the great Cambrian Lagerstätten. A million years later, here we are in northern Greenland. At that time, Sirius Passet was located in an area that is today polar and often frozen. The configuration of continents and the global climate were quite different about 517 million years ago. However, this environment was colder than the tropical regions of the Cambrian, with an average water temperature around 10 to 15 degrees Celsius, much milder than present-day Greenland. The Lagerstätte of Sirius Passet was formed in a shallow marine basin, offering an interesting contrast to the warm waters of Chengjiang. Sirius Passet fascinates with the diversity of its fauna, preserved in fine shales revealing exceptional anatomical details. Its fauna resembles that of the Chinese Cambrian, but presents adaptations suggesting a different lifestyle linked to cooler waters and specific nutritional input. Some animals there likely had a metabolism adapted to these lower temperatures, allowing them to survive, where others struggled to thrive. Among the emblematic fossils of Sirius Passet are vermiform organisms, similar to predatory worms, equipped with robust mouthparts to capture small invertebrates. There are also unique arthropods, perhaps equipped with articulated plates, allowing them to move over loose sediments. The exceptional preservation sometimes reveals their musculature, even the shadow of digestive organs, thus offering a valuable glimpse into their internal anatomy. Although the species diversity there is less than in some other deposits, Sirius Passet plays a crucial role in enriching our understanding of Cambrian biodiversity. It demonstrates that at 517 million years, life was not limited to warm tropical waters, but also adapted to harsher environments, exploiting diverse ecological niches. In this sense, Sirius Passet is a key link, revealing that the marine life's revolution extended to various regions, including cooler zones. Let's continue our journey through time to Kangaroo Island, Australia, where the Emu Bay Formation, about 515 million years old, offers a fascinating glimpse of a major evolutionary advancement, the sophisticated development of visual systems. Imagine this shallow continental shelf, bathed in warm waters around 25 degrees. The sediments deposited here trapped remarkably well-preserved creatures, some of which possessed compound eyes, as complex as they were effective. A real asset in a world where predation was becoming a crucial issue. Emu Bay is famous for revealing fossils of animals with faceted eyes, very similar to those of modern insects and crustaceans. These compound eyes, composed of many small units called ommatidia, offer a wide field of vision and great sensitivity to movement. Some Cambrian predators had an impressive number of units, making them very efficient at tracking their prey. This advancement likely spurred an evolutionary race, with prey developing camouflage and defense, and predators honing their senses. Among the star animals of Emu Bay, a large predatory arthropod, sometimes related to radiodonts, possessed eyes with many lenses, giving it a highly developed vision compared to other poorly sighted creatures. These fossils sometimes show fossilized pigments, tiny molecules that color the eye and detect light. Scientists deduced that sight was crucial for quickly spotting prey or rivals from a distance. Emu Bay also reveals the diversity of marine organisms, primitive crustaceans, sponges, brachiopods, and other fossils often protected by plates or spines. All testify to a dynamic life where competition drove evolutionary innovations. Beyond its advanced eyes, the Emu Bay Formation marks a key step in the biological arms race, an essential milestone for animal evolution. We now move up to Canada, in the Rockies, to discover the Burgess Shale, dated to about 508 million years ago. This fossil site, one of the most famous in the world, owes its fame to exceptional preservation, allowing detailed study of the morphology of many marine creatures. Originally, it was a submarine slope where flows of calcareous or silty mud regularly buried living organisms above. The Burgess Shale is renowned for its exceptional diversity, with hundreds of species. There are arthropods with strange shapes, some with long spines, others with multiple limbs. Other enigmatic fossils represent extinct groups with no direct descendants today. This abundance of evolutionary experiments shows creatures that did not always find their place in the tree of life and became extinct, leaving only their fossil traces. Besides the diversity, a recent discovery has revealed intriguing chemical residues. Using spectroscopy, researchers detected copper in the fossilized tissues of the Burgess Shale, suggesting a circulatory system based on copper, like hemocyanin in current mollusks and arthropods. This discovery confirms that as early as the Cambrian, nature was testing different ways of transporting oxygen, essential for an active metabolism. The burial conditions in the Burgess Shale, with sudden flows of fine sediments, have frozen life in a geological snapshot. Imprints of skin, muscles, or digestive systems nourish detailed research, rich in insight. The Burgess Shale is a true open book on the marine diversity of more than 500 million years ago, and a key pillar for understanding evolution in the Cambrian. 3 million years after the Burgess Shale, here we are in Utah, in the Margam Formation, about 505 million years old. At that time, the region was under a marine environment, far from today's aridity. It formed a shallow continental basin, bordered by reefs and crossed by multiple marine currents. The water was relatively warm, around 20 to 25 degrees, a favorable climate for marine life. This site fascinates specialists because it has revealed a vertebrate named Eutahaspis. A vertebrate is an animal with a backbone or a similar internal structure. The presence of such an organism at this time is crucial for understanding the emergence and distribution of the first vertebrates, a group that later includes fish, amphibians, reptiles, birds, and mammals. Eutahaspis was an elongated aquatic creature without evolved jaws, with a simple skeleton, probably composed of cartilaginous tissue and rudimentary mineralized elements. Thanks to the remarkable preservation of the Marjum Shale, researchers observed the details of Eutahaspis's body structure. They distinguish, for example, the arcs supporting the gills, revealing its underwater respiration. The segmented chevron-shaped musculature is visible in some fossils, offering a valuable clue about its movements to hunt or flee. The discovery of Eutahaspis illustrates the spread of chordates in the Cambrian seas and marks a key milestone in the evolution of our animal lineage. Alongside numerous marine invertebrates, another adventure was unfolding, that of animals with internal skeletons, future dominators of the planet. Long overshadowed by the Burgess Shale, the Marjum Formation is nonetheless essential for understanding the multiple paths life took during the Cambrian. A few million years later, we find ourselves in China, at the Linyi site. This site bears witness to a warm and nutrient-rich Cambrian marine environment, conducive to a diverse and astonishing fauna. The rock layers have preserved arthropods with specialized claws or limbs, the grapplers. These arthropods had anterior limbs adapted for seizing prey or clinging to algae and rocks, ensuring their stability in the face of currents. Their anatomy reveals an adaptation to predation and turbulent environments. These robust and segmented claws could exert enough pressure to immobilize small crustaceans, worms, or other smaller arthropods. The fineness of the fossil preservation at Linyi allows for the observation of detailed appendage structures, including muscle impressions and ligament insertions. These clues provide rare insights into their mobility. One can infer different hunting strategies. Some would swoop down on their prey in a flash, while others adopted a slow and stealthy approach to surprise their victims. The Linyi fauna was not limited to the formidable grapplers. There were also more discreet species, protected by thin shells, likely disadvantaged against such predators. The marine flora, composed of algae-forming mats on the shallow sea floor, offered shelter and food. This site illustrates how, even at that time, morphological specialization was vital for survival. Our last stop takes us to southern China, to the Kaili Formation, about 497 million years old. This site marks a turning point in the evolution of trilobites, these emblematic arthropods of the Paleozoic, already diversified for millions of years. Kaili reveals a major innovation, the appearance of filtering behaviors, allowing the capture of food particles suspended in the water. What does this lifestyle mean? Instead of hunting or foraging in the sediment, some trilobites anchored themselves or moved slowly, using their legs or specialized mouth parts to filter water and extract microorganisms and nutritious debris. Today, this mode exists in filter-feeding whales and some crustaceans. For these trilobites, it was an efficient way to exploit the abundant plankton. The regular deposits of limestone mud at Kaili facilitated the fossilization of invertebrates often found together, forming real marine communities. The filtering trilobites probably cohabited with sponges, brachiopods, and algae, each occupying a precise niche. Thus, the filterers exploited a specific stratum, while predators, burrowers, and grazers shared other resources. The emergence of filtering trilobites illustrates a key diversification. Changing diet avoids competition and opens the way to new specializations. Kaili reveals that at the end of the Cambrian, evolution continued to innovate, showing the remarkable adaptability of trilobites through their numerous anatomical transformations. Here is our overview of the major Cambrian Lagerstätten, witnesses to a marine life in full explosion. Each Lagerstätte provides a key to understanding the Cambrian, revealing frozen moments when animal life was taking shape. These deposits, studied with advanced methods, all aim to capture the diversity, evolutionary strategies, and ecological upheavals of this era. Silent yet powerful witnesses, they remind us of the richness and fragility of ancient life, and the immense fortune of still being able to explore its fossil remains. Research on Cambrian fossils is experiencing a spectacular surge thanks to new scientific techniques between 2023 and 2025. These modern methods provide unprecedented insights into organisms hundreds of millions of years old. Imagine researchers moving from an excavation site to a synchrotron, then to a geological dating laboratory, and finally consulting astronomical data to better understand ancient climate cycles. These approaches complement each other to revisit the history of life in the Cambrian with unparalleled precision. This section will introduce you to the secrets of 4 main methods with the wonder of an explorer on new land. The first method, 3D synchrotron tomography, allows for observing fossils without damaging them. The second, in situ Raman spectroscopy, detects traces of fossil molecules and pigments. The third, CATIM's uranium-lead, dating on ash layers, offers exceptional chronological precision. Finally, astrochronology correlates astronomical cycles with sedimentary deposits to analyze the rhythms of environmental transformations. These approaches have revolutionized recent paleontological studies, providing precise results and symbolizing scientific modernity applied to ancient geological times. Let's start with 3D synchrotron tomography. In paleontology, fossils are often very fragile and risk being damaged during a traditional dissection or sectioning. Moreover, some essential details such as soft tissues or internal microorganisms often escape fine sections or optical microscopes. This is where the synchrotron comes in, a large particle accelerator where particles circulate at high speed in a magnetic ring, producing highly energetic radiation. This radiation penetrates matter with a resolution reaching a micron, a thousandth of a millimeter, thus revealing internal details invisible otherwise. "3D tomography enables the study of fragile fossils without damaging them. A small rock fragment is placed on a rotating stage, and the system captures hundreds of images from all angles. Software reconstructs a 3D model, allowing exploration of the interior without physical cuts. This technique is essential for examining fragile structures like nerves or fossil vessels, often destroyed by mechanical dissection. In sites like the Burgess Shale, it reveals rare tissues and studies the internal anatomy of ancient species, from circulatory channels to nerve segments. This method has become indispensable in paleontology for exploring the intimacy of fossils. The second method, in situ Raman spectroscopy, focuses on the chemical composition of fossils and surrounding rocks. It is based on the interaction between a laser and molecular bonds. When a laser beam illuminates a sample, a small portion of the scattered light changes wavelength according to the present molecules. This change creates a Raman spectrum, a sort of unique chemical fingerprint. Thanks to this technique, researchers identify minerals, organic remains, and pigments directly in the field or in the lab without altering or dissolving the fossil. In situ means that the technique is applied directly on a rock slab or fossil, often without complex manipulations. For example, in situ Raman spectroscopy allows detecting eye pigments in the compound eyes of ancient arthropods. This non-destructive method uses a laser to obtain a chemical signature and map the distribution of pigments or minerals on the fossil surface. When combined with 3D synchrotron tomography, it reveals both internal structure and chemical composition, enhancing our understanding of the anatomy and lifestyle of specimens. Now, let's move on to C.A. Timms uranium lead dating on ashes, a key method for the Cambrian chronology. C.A. Timms, chemical abrasion, thermal ionization. Mass spectrometry, by definition, refers to thermal ionization mass spectrometry preceded by chemical abrasion. It is primarily used to date zircon crystals, a common mineral in volcanic ashes. These ash layers, often trapped in Cambrian marine sediments, constitute precise temporal markers. By forming between fossiliferous layers, they record the exact age of the volcanic eruption. Uranium lid dating relies on the decay of uranium to lead in zircon crystals. By measuring the amounts of uranium and lead, the time elapsed since the crystal's formation is calculated. The C.A. Timms method adds a chemical cleaning to remove contaminated areas, followed by ionization in a mass spectrometer to precisely measure uranium-lead ratios. This method, with a temporal resolution of less than 1/10 of a million years, allows for precise dating of Cambrian events, such as the appearance of specific faunas or local extinctions. Thanks to C.A. Timms, several layers of the Meow-Lingian have been accurately dated, providing a precise chronology for Cambrian events. The fourth technique, astrochronology, shows how astronomical cycles influence sedimentation and the geological record of ancient periods. This discipline studies and correlates Milankovitch cycles, which correspond to the periodic variations of Earth's orbit and its tilt. These cycles include orbital eccentricity, axial obliquity, and precession, which modulate the distribution of solar energy, causing climatic oscillations. While their role is well known for recent glacial periods, geologists have also demonstrated that they leave an imprint in very ancient sedimentary deposits. Astrochronology involves observing the alternation rhythms in strata, such as light and dark layers or repeated chemical variations. By linking these frequencies to Milankovitch cycles, one can accurately date and correlate geological formations. Its resolution often reaches a few thousand years, useful for archives of hundreds of millions of years. In the Cambrian, it helps identify phases of oxygenation, sea level fluctuations, or alternations in biological productivity. Astrochronology complements radiometric dating like C.A. Timms and classical stratigraphy, offering subtle chronological markers. These methods never work in isolation, but complement each other to provide a comprehensive view of the same subject. For example, the discovery of a new fossil species in a layer dated using the C.A. Timms method allows for knowing its exact age. Synchrotron tomography reveals its internal anatomy. In situ, Raman spectroscopy confirms the presence of sensory pigments, and the study of Milankovitch cycles verifies if certain biological adaptations correspond to cyclical environmental variations. Together, they allow for a complete portrait of the fossil, its age, anatomy, chemical composition, and environment, helping to better understand the tree of life, especially during periods rich in innovations like the Cambrian. These techniques are constantly evolving, improving rapidly between 2023 and 2025. Synchrotrons gain in brightness and stability, enhancing image quality. In situ, Raman spectroscopy benefits from more precise lasers, increasing sensitivity even on very small or altered samples. The C.A. Timms uranium lead protocol, once reserved for a few laboratories, is now spreading to many geological centers. As for astrochronology, It takes advantage of advances in astronomical models and vast geological databases, making it easier to compare cycles between continents. These advances are revolutionizing the study of Cambrian archives. In the past, scientists searched for index fossils and used rudimentary dating methods. Soft tissues, such as nerves or eyes, were often ignored. Today, synchrotron tomography clearly distinguishes the shell from the internal structure. Raman spectroscopy reveals chemical composition, and CA-TIM's uranium-lead dating anchors these discoveries in a reliable chronology. Astrochronology provides a framework for understanding rapid environmental variations. Together, these methods form a precise puzzle, enriching the overall vision of evolution. To illustrate this synergy, imagine a group of scientists arriving at an outcrop where a thin layer of volcanic ash lies between fossiliferous levels. They collect samples, isolate zircon crystals in the laboratory, which they prepare through chemical abrasion for uranium-lead dating via mass spectrometry. Meanwhile, a fossil block containing a Cambrian arthropod is taken to a synchrotron. 3D tomographies are performed to examine its visual apparatus, suspected to preserve mineralized nerve structures. A reconstruction in digital images reveals pigmented ocular channels. The researchers then apply in situ Raman spectroscopy to confirm the presence of a pigment similar to melanin. To determine if these pigments indicate a climatic adaptation, they consult astrochronological data indicating precise temperature and light cycles affecting shallow waters. Combining this data, they hypothesize that this species developed more advanced eyes, possibly in response to a rapid restructuring of the food chain linked to marine environmental fluctuations. Each method contributes a piece to the puzzle, fitting into the grand geological mural. In the coming years, these methods are expected to progress. The 3D synchrotron tomography will benefit from more powerful facilities, allowing faster rotations and better reconstruction. In situ, Raman spectroscopy could detect various structures such as proteins or fossil lipids. For CA-TIM's uranium-lead dating, the hope is to reduce contamination for greater precision. Astrochronology will benefit from more precise spatial data to refine ancient solar system models. These advances will strengthen a high-precision geology and paleontology, allowing the exploration of fossils in their anatomy, chemistry, chronology, and environment with growing certainty. Behind every spectacular fossil displayed in a museum often lie these cutting-edge techniques. They reveal unsuspected structures, refine the age of layers, unveil unknown climatic dynamics, and propose more complex evolutionary scenarios for the Paleozoic. The period from 2023-2025 is pivotal. After a decade of innovations, researchers now have tools to re-examine old samples and explore new terrains. Geology, chemistry, particle physics, and astronomy unite to fill the gaps in the history of life on Earth. In the current effervescence of paleontology, the boundaries recede and mysteries fade. Researchers reconstruct the anatomy of species extinct for eons as if they were projected under a beam of light. Ocular pigments, fossilized nerves, microvascular structures, precise dating between distant deposits, and climatic signals from Milankovitch cycles redraw the history of Cambrian biodiversity. Far from being anecdotal, this wave of discoveries serves as a model for other periods, showing that it is possible, even in the oldest records, to distinguish the true from the supposed, to reassess events, and to enrich our understanding of evolution. At the beginning of the Cambrian, marine life was beginning a spectacular diversification. Between 2024 and 2025, several major discoveries shed light on this ancient chapter. We will first explore the proto-mollusk Schistonia aculeata from the fourth stage of the Cambrian, then two recent chordates from the Drumian, Utahaspis, and a supple chordate from the Marjum Formation. We will also address the isotopic recalibration of the Drumian in the Yangtze region, introducing a substage based on a negative excursion of D13C. This journey takes us into a world where the oceans reveal the beginnings of cephalopods, the first vertebrate skeletal structures, and environmental fluctuations visible through the chemistry of rocks. First stop, the discovery of Schistonia aculeata in the fourth stage of the Cambrian, approximately 514 million years ago. At that time, marine life was already abundant, but some primitive fossils were difficult to connect to major animal groups. The announcement of Schistonia in 2024 in China's Yunnan province garnered significant interest. This region is known for its exceptional fossils, preserving rare soft parts. Schistonia reveals a spine-bearing proto-mollusk form, suggesting that the early stages of mollusk evolution already included defensive adaptations and advanced body structures. The term proto-mollusk refers to a probable ancestor of modern mollusks, squids, octopuses, gastropods, bivalves. It generally possesses a muscular foot, used both for movement and for temporary anchoring to the substrate. In Schistonia aculeata, a series of external spines suggest protection against predators present in this ecosystem. Although it does not yet have the spiral shell of gastropods, other traits, such as the shape of the foot, show an essential evolutionary link. Thanks to synchrotron tomography, internal analysis has revealed the structuring of soft tissues, indicating specialized organs for respiration and nutrition. In a warm marine environment, with a temperature between 20 and 30 degrees, Schistonia evolved near the bottom, possibly grazing on biofilms or small organisms. Its spines, already well developed at the fourth stage, testify to the strong pressure exerted by the early Cambrian predators, such as some aquatic arthropods. This competition favored the appearance of protective armors, exoskeletons, and mineral structures, marking what specialists call the Cambrian Explosion, a period when many body plans were formed in a short time. A few million years after Schistonia aculeata, the Drumian starts, between 504.5 and 505 million years ago. During this period, the American fossil record reveals its treasures, notably in the outcrops of Utah. Two discoveries, announced by the Natural History Museum of Utah and the site phys.org, have reignited the debate on the early origin of our evolutionary group. Utahaspis is a chordate essential for understanding the origin of vertebrates. Chordates possess a dorsal chord, a flexible axis that structures their body. In modern vertebrates, this chord gradually transforms into a segmented vertebral column. In Utahaspis, the first signs of this organization are observed: V-shaped muscle segments and a more rigid dorsal structure than in other Cambrian fossils. These fossils come from shallow marine strata, where the water, according to paleoclimatic estimates, reached about 25 degrees C. This varied environment housed not only arthropods and sponges, but also chordates more evolved than previously thought for this period. The second discovery, in the Utah region and dating from a nearby period, concerns a soft-bodied chordate found in the Marjum Formation, known for its exceptional preservation, sometimes including soft tissues. According to a release echoed by phys.org, this animal is the first confirmed Drumian vertebrate in North America. To be qualified as a vertebrate, There must be internal ossification, or at least segmented cartilaginous skeletal elements. Even though these beginnings of ossification are subtle in the Cambrian, a meticulous study of certain imprints reveals an emerging vertebral structure. This soft-bodied Drumian chordate possesses visible gill pouches, a sign of a sophisticated respiratory system to capture dissolved oxygen. In the middle, Cambrian chordates already occupied various marine environments, surpassing the basins of East Asia, where Milocunmingia and consorts were found. This broad distribution illustrates their rapid dispersion and adaptation. Each new fossil fills gaps in the evolutionary tree by showing how segmented musculature, internal support structures, and respiratory pathways converge to form vertebrates. Parallel to these discoveries, another project focuses on the chemistry of Drumian rocks on the Yangtze platform in China. In 2025, a publication relayed by Science Direct describes an isotope recalibration of carbon isotopes. It involves studying the ratio between carbon-13 and carbon-12 isotopes, called delta-13C. A rapid decline of this indicator indicates a reduced proportion of carbon-13 relative to carbon-12 over a short geological period. Why is this isotopic gap so important? These variations reflect major disturbances in the carbon cycle, linked to volcanic activity, biological productivity, or the decomposition of organic matter. A decline in delta-13C can indicate a massive input of light carbon, for example from accelerated decomposition, or warming, promoting the release of carbon dioxide. These fluctuations change marine conditions, dissolved oxygen, plankton distribution, and trophic interactions. They directly influence the evolution and diversification of organisms, notably the chordates and proto-mollusks mentioned. The isotopic recalibration of the Drumian in the Yangtze, a marked negative excursion, constitutes a precise marker to refine the Cambrian chronological scale. Researchers are even considering defining a new substage based on this pronounced geochemical signal, linked to an ecosystem reconfiguration. This substage would offer a global reference for comparing similar geological sections, enabling an evaluation of whether distant paleontological events, like the appearance of Utahaspis in Utah and the diversification of chordates in the Yangtze, coincide with these global isotopic fluctuations. The link between internal biological innovations and environmental changes remains a key focus in paleontology. Evolution does not occur in isolation. It is stimulated or hindered by its environment. An increase in carbon dioxide can alter water temperature and nutrient availability, changing species distribution. Conversely, an ecological shock pushes some lineages to develop new biomechanical, physiological, or behavioral strategies to survive. In the Cambrian, the almost simultaneous appearance of powerful predators was a major driver of the explosion of body forms. Isotopic analyses illuminate the chemical and thermal backdrop where this evolutionary scenario played out. The discoveries of 2024 and 2025 expand our view of the early stages of complex animals. Biological and geochemical events demonstrate a dynamic Cambrian marine universe conducive to the emergence of major lineages for subsequent eras. Through cutting-edge methods, paleontology surpasses the superficial observation of damaged bones to become an integrative science, blending Earth's chemistry, comparative morphology, and the study of ancient environments. Thus, each new discovery, whether it is a spiny proto-mollusk or an unknown chordate, can revolutionize our understanding of the origins of animal life. Ultimately, the Cambrian, far from being a mere primitive age, appears as an era of major upheavals. Lineages we thought were late were already present, sometimes discreetly, in these ancient seas. The alliance of paleontology and geochemistry places these creatures back in a dynamic context. Thanks to Shishania aculeata, Utahaspis, the flexible chordate of the Marjoom Formation, and the negative excursion of Delta 13c, we now have a clearer panorama of this evolution. Each new excavation will enrich this history, showing that complex life was forged on the fine edge of Earth's physical and chemical balances.